Hello, good morning, everybody. Bom dia a todos. Welcome to BioISI Day. We have a rather unusual format, but this actually has become our usual format in the recent days. So we have a rather complete program for today. I hope you will enjoy it. But if you're not able to participate, we will make available the program in the internet through YouTube, so you can watch it later. So without further ado, I'm going to start showing and sharing with you some of the research and some of the services and other activities that we have at BioISI. I'm going to focus particularly on the most recent activities that occurred during this year, 2020, the pandemic year. It will be, I'm sure, known for years to come. So at BioISI, we have the vision to be a recognized leading international systems biology center. And our mission is to explore integrative sciences. Our research and technology is focused on biosystems and integrative sciences, targeting global societal changes and industrial competitiveness in biology and medicine. So at BioISI, we have 5 key missions. The central one is, of course, research, but we also have facilities and services, advanced training, knowledge and technology transfer, and also development of technology and instrumentation. Starting with research, we have 5 main areas of research, which we call thematic lines, and these include bioinformatics, and we focus on digital systems biology and systems modeling, biophysics, innovative technologies, magnetic nanoparticles, biological chemistry, bioactive molecules, cellular bioenergetics, biomedicine, mechanisms of disease and personalized therapies, biotech, sustainable agriculture, and sustainable environment. And this research is carried out by 8 very large research groups with multiple PIs in each group. And we have 3 flagship projects. Our flagship projects are cystic fibrosis, enabling technologies, grapevine, and wine. And the outcomes of our research actually contribute to the 17 United Nations Sustainable Developmental Goals. Some of them with a greater focus, but also some others with some horizontal skills that contribute to these goals. In total, BioISI has 344 members, not all with PhD, of course, and this is the distribution of the members in our center. So we have a significant proportion of students, both master and PhD students, young PIs who are starting their careers, more established scientists, and senior PIs. We also have management staff and other staff. This is the gender distribution. I think we are pretty well according to the EU standards. If anything, it's unbalanced towards the female side. As faculty members, we have more or less 50-50 non-faculty and faculty members. These are some of BioISI key performance indicators, or KPIs. So these are the numbers for 2020, and here you can see the evolution since 2015. So these are the total numbers for 2020, as I said, and these are the members I mentioned. Here we have a total publications, indexed publications, close to 200. This is still just the three first trimesters of 2020. And we have an average impact factor of the top 25% papers of 7.2 and about 3 million euros per year of competitive funding. Still a little bit of bibliometrics. This is not, of course, everything, but it is also important to note. So these are the papers, and here in the columns in gray, you can see the papers within the first quartile. And I must stress again that these numbers for 2020 are just the first 3 trimesters. And this is the evolution of the impact factor of the top 25 publications. And here you have examples of what these publications, these top publications, which journals they are in. So breaking down the funding along the years, this is pretty much the competitive funding that BioISI researchers have been able to attract. And this is the distribution from governmental funding. This is mostly FCT, 13% of private funding, companies, industry, and other private sources. And here you can see in orange funding from European Commission. As to technology and instrumentation, so part of the mission of BioISI is actually to develop novel technologies, such as this example of a variant of atomic force microscopy, which is shown here, a force feedback microscopy, or this computational tool for precision medicine in cancer, which is part of an European project. Facilities and services. You will hear more after my presentation about facilities and services in detail, because we believe this is of interest not just for BioISI researchers, but of course all members of COOL and also in the Lisbon area and maybe also beyond. So the goals of BioISI facilities are providing state-of-the-art equipment and user support to BioISI researchers to reach the next level of their research, so they can apply to competitive funding internationally, so European projects, Horizon 2020 or Europe 2020, ERC grants, and so on. Turning BioISI into a societal player for research infrastructures in Lisbon region, and making excellent services available to society to innovate faster and more effectively. So in terms of facilities, we have bioimaging, which actually we share with other centers at FCOOL. So this is the FCOOL microscopy facility. We also have a high throughput facility, which includes not just robots as shown here, but also automatic stage microscopes. We have mass spec for metabolomics and proteomics, and genomics facility, which is actually doing 4th generation sequencing. Besides the facilities, we also have structures for our researchers to carry out their activities. These include common animal cell culture, plant house, radioactive room, microbial and fungal biobank, AFM-related techniques, and computing, which is actually hosted at FCOOL's Informatics Center. In terms of advanced training, we have a very successful BioISI PhD program in place with a focus on systems biology, which was allocated 55 PhD fellowships. It's only based at FCOOL, so with no other institution, but a significant part of these fellowships are actually international. So we collaborate with many key international institutions for these fellowships. So they have lots of activities on their own, things like retreats, PhD day, and of course, thesis discussion, which this year has actually had a different format. So this PhD program in systems biology already produced outcomes, and until now, 63% of the students out of the 32 who actually have finished their time, 20 have completed their thesis. And out of the 20 who completed the thesis, 65% already started a new position 1 month after graduation as postdocs, lecturers, scientific officers in industry. And these 20 graduates contributed to 68 publications during their PhD work, and they are first authors in 37 out of these 68 publications. So 21 do have a focus on systems biology, modeling, or advanced biomedicine, novel models, and personalized medicine. So this is just the students from BioISI's PhD program, but among all BioISI PhD students, 51 completed their thesis in the period between 2015 and 20. So this gives an average of 9 theses completed per year in those 5 years. And BioISI has currently 76 PhD students. Besides, they also have 141 master students in this, we had in these 5 years, and they have completed about 24 theses per year. So still on advanced training, we have 3 cycles of seminars, the postgraduate seminars and PhD student seminars, which they organize on their own, the BioISI Research seminars, which are usually provided by BioISI PIs, and we also have an international research seminar program with key researchers from different parts of the world. Still on the topic of advanced training, we have organized hands-on workshops and summer schools with lots of activities in our own labs. And we did have a BioISI postdoc program, which we are planning to restart again. And in the past, we had 4 postdoctoral fellowships for 2 years each to carry out multidisciplinary cross-group projects on BioISI, thematic lines, and strategic goals. So these fellowships were actually aimed at reinforcing and strengthening the interdisciplinary research which goes on at BioISI. On knowledge and technology transfer, so-called KTT, we have a long-standing role, namely by the microbiology and biotechnology group, in supporting small and medium enterprises incubated at tech labs. So this is Schools Innovation Center. And in the past 4 years, BioISI members registered 5 patents and 14 computational models. In terms of bioeconomy outputs, we have a focus on the sea, from where we extract biocompounds for multiple purposes, health, food, cosmetics, industry, and so on, and innovation solutions to 28 startups, actually hosted at tech labs for most of them. We actually collaborated in a co-lab on smart farms, and of course, I'm sure you all know about the school center of COVID tests, in which BioISI actually has a key role. These are some examples of key contracts with industry. We collaborate on interface of software with health, on food industry for smart wines, smart farms, the co-lab I already mentioned, and we have been collaborating with industry, in this case AstraZeneca, for screening of bioactive compounds in our high throughput screening unit. Knowledge transfer is also communicating and giving things back and knowledge to society. So besides the strong interaction with companies, not just in campus, but also external and multiple industry contracts, we also have several national and international prizes and awards, which actually recognize our scientific merit. And these two are particularly interesting because we heard about them yesterday. And these are two very important and prestigious awards given by CNOIV. And two different groups in BioISI were actually awarded these awards. Also, transfer of knowledge is also communicating to the society, and we participate in several activities to communicate what the outcomes of our research to society, not just in this European work, European Week of Research, but European Researchers' Night, besides many actions at schools. And this was the summary I wanted to present to you, and I wish you a very interesting day hearing about what goes on in terms of research at BioISI. And thank you very much for your attention. I believe my colleague, Rui Malhó, will provide some overview on the facilities, and in some of detail, the heads of those facilities will also provide information about the services which are offered and what we can offer to the society on this regard.
Okay.
BioISI is an interdisciplinary research centre that aims to understand and address biological questions using integrative systems approaches at the vanguard of life sciences research. BioISI's research is based on Faculty of Sciences of the University of Lisbon in association with the National Health Institute Lisbon, Minho University and UTAD. At our laboratories, we work to understand the complex properties that cannot be predicted from studying the isolated parts of a biological system. The vision of BioISI is to pursue cutting-edge research to become a leading centre at the forefront of research, development and innovation.
Interdisciplinarity is of key importance to BioISI. BioISI actually encompasses several scientific areas, and this is the focus of our institute.
BioISI offers a unique multidisciplinary environment that fosters creative thinking to solve problems through integrative approaches, gathering researchers with backgrounds in biology, chemistry, physics, and computer science. BioISI is training the next generation of scientific leaders in biosystems and integrative sciences. BioISI's objectives are implemented along 5 thematic lines: biomedicine, biotechnology, biological chemistry, bioinformatics, biophysics. The core of BioISI's research activities is driven by 3 flagship projects: crop product improvement and contributions to bioeconomy, grapevine and wine. Systems approaches to rare diseases, cystic fibrosis and neurodegeneration. Enabling technologies to boost innovative research in biological systems. BioISI is a privileged space for research, development and innovation, enabling the creation of novel technologies and instrumentation, with state-of-the-art facilities open to internal and external collaborations, providing services for both academia and industry. At BioISI, knowledge and technology transfer is a priority. BioISI has a team of dedicated staff to create value from research to society. Teaching and training. BioISI hosts the BioCys PhD programme on systems biology since 2014, enrolling 55 promising young scientists. It provides a stimulating environment for postgraduate training by actively promoting the mentoring of young PIs for independence, career development, international workshops, seminars. An important aspect of BioISI activity is to engage with societal challenges and ensure that the relevance of our work and research outcomes is perceived by the public and contributes to improved policies. We are now more than 250 researchers at BioISI. Be a part of our journey.
Hello and welcome to BioISI annual meeting. My name is Rui Malhó and I'm the coordinator of BioISI facilities. At BioISI, one of our key points is to ensure that we provide services and also access to all modern equipment that we have available and which we organize in facilities. We also have what we design or call as infrastructures, which are key equipments that are able to be used by everyone on the basis of a booking, but do not consist on the instrumentation parts and the access to technical services that we provide in the facilities. So infrastructures that we have available for the use of our members and also the outside community include plant growth facility or growth chambers, animal cell culture, computing, and also atomic force microscopy. By facility, we designate something like the microscopy facility, the genomics facility, and the mass spec facility, where alongside equipments that are able to produce results on these methodologies, we also have full support, full technical and staff support to ensure that our members have the best of the research and the interdisciplinary approach that we aim here at our research unit. BioISI Genomics has been a great assistance to the Faculty of Science in our fight against COVID, and that will be shown later on by the responsible of the facility, currently Ricardo Dias. Okay? And the mass spec facility will be shown to you later on by the responsible, Luísa Serralheiro. I'm also running the microscopy facility, which, as you can see here, includes full booking system for internal and external users, okay? And which includes, in terms of equipment, both inverted microscopes for epifluorescence, wide field microscopes, confocal microscopes, 2 confocal microscopes, one more devoted to morphology analysis, and the other one to high throughput experimentation. The high throughput sequencing unit is also part of BioISI and facility that we are implementing, and that will be shown you also by the responsible, which is Hugo Botelho. This high throughput facility is now part of PT Screen network, and our microscopy facility in general is part of the Portuguese platform for bioimaging, which has now been recognized as a partner of Eurobioimaging. As I was saying, alongside with these main equipment for the epifluorescence, we also have functioning scanning electron microscope and transmission electron microscopes, which are operated by a cool faculty of science technical staff. All these equipments that we have available, as I said, can be booked through an agenda system, and for that you just need to have access to our website, which you can have to time course analysis and 3D analysis of embryo development, plant organ development, and anything to do with image reconstruction and analysis over time and space. While the high throughput screening, as you will see, will be more devoted to types like pharmacological analysis and the screening of large compounds and mutations that can be of interest to one of our thematic lines and to the research topics that we have available. In the past 2 years, the BioISI microscopy facility has directly produced more than 20 research peer-reviewed manuscripts and involving more than 40 authors, members of BioISI. Aside that, we also have approximately 30% of external users from the faculty, so away from the faculty, that are involved and use this facility. The topics that have been covered by our research goes from animal to plant cells to microorganisms, namely bacterial development, plant cell development using model systems like Arabidopsis and our flagship projects like Vine and Quercus research, to animal diseases, human diseases like cystic fibrosis, to cell cycle regulation and oncobiology. Alongside with the equipments that we have available, the microscopy facility also provides training for young postgrads, postdocs, and has been also involved in EMBO courses where some of our staff has been involved here in Lisbon and also at other labs of the Eurobioimaging network. Alongside with these training courses, we also provide training to users that want to do image processing. So we have modules on image analysis, and this is part also of our BioISI PhD program. And another service that we have at the facility is the use of 3D printing in order to provide the members with adapters for example, cell phone microscopy, to couple old devices that were analog to digital devices, and so to increase the automation of the equipment. The image analysis capacity is obviously related to our computing facility and to our computing infrastructures, which will be explained to you also by our colleague Miguel, which is now the coordinator of the bioinformatics thematic line.
high-throughput screening facility is a research infrastructure for spectroscopy and image-based screening hosted by BioISI. It supports over 40 users and provides over 5,000 hours of instrument access each year. The facility provides user training, consultancy for assay design, as well as open access to state-of-the-art equipment for sample preparation, bioimaging, microplace-based measurements, and data processing for users from BioISI and other institutions. High-throughput screening encompasses a collection of technologies enabling the efficient acquisition of spectroscopic or image-based features from specimens treated with a diverse set of compounds, DNAs, or RNAs. The screening campaign may target a library with many thousands of treatments, and therefore requires specialized equipment, staff, and expertise. The facility has an important focus on microscopy. It features automated wide-field and confocal systems with high-content capabilities, which can image biological specimens laid out in multi-well plates in 2 or 3 dimensions. Both system types have lifestyle imaging capabilities. High-throughput screening requires a specialized infrastructure to assure efficient and reproducible sample preparation. The facility provides such equipment in terms of the seeding of eukaryotic cells, automated liquid handling, reagent pipetting, and plate washing. Data acquisition is performed on automated microscopes or plate readers. The most challenging screening campaigns deal with complex phenotypes. These high-content screens rely not only on robust image analysis algorithms, but also on strong data science and statistics expertise to appropriately handle the data and pinpoint relevant discoveries. The facility supports its users in these tasks by providing bioimage analysis, data exploration, and software development services. The facility is staffed by a team with complementary expertise, composed of a manager, a microscopy technician, and an image analysis technician. The high-throughput screening facility is well integrated within the national roadmap for research infrastructures. We are members of PT OpenScreen, the national infrastructure for genetic and chemical biology, and PBI, the Portuguese platform of bioimaging. In each case, the facility is on track towards integration into the corresponding European research infrastructures, EU OpenScreen, and Eurobioimaging. Facility users benefit from instrument and bioimage analysis training on demand. Furthermore, the facility has regularly hosted a hands-on workshop on high-throughput microscopy, a week-long course which gives its participants the opportunity to learn about assay design, imaging, and high-content analysis. Some of the facility's most relevant scientific outputs relate to the genetic disease cystic fibrosis. Facility users have performed screens on natural products or compound libraries aiming at discovering or repurposing molecules with biomedical relevance. Functional genomic screens to identify genes and gene networks which may be targeted for therapy, and lifestyle screens addressing basic cell biology or personalized medicine. The facility has also produced workflows for high-content image and data analysis. The high-throughput screening facility serves BioISI members, the academic community, and corporate users. Contact us for more information.
Welcome to the BioISI mass spec facility. This is composed of a chromatograph and a mass analyzer. The chromatograph is an UHPLC and mass equipment uses an electrospray ionization process. It is a QTOF. The chromatograph is equipped with 2 different types of columns, a reverse phase and an ilic, so either hydrophobic and hydrophilic compounds can be separated with this equipment. The mass equipment allows the determination of exact mass and also allow the mass fragmentation for helping in the identification of the compounds. The analysis can be done in negative and positive ionization mode. The UHPLC can accommodate more than 50 samples and it injects 24 hours a day. We can detect compounds till 1 microgram per milliliter and masses from 50 to 148 kilodaltons. This facility has already done several different types of work. Identification of small compounds in algae extracts, in medicinal plants, in grapes, in wine, biocide detection in waters, and analysis of several drugs. It has also determined exact masses for new compounds obtained by chemical synthesis. Our new topic of development now is metabolomics. With metabolomics, we can study whatever we want, whenever we want to find biomarkers. So for this moment, we started studying the effect of aqueous extract from algae on liver cell metabolites. The objective is to find biomarkers for the effect of this type of metrics. We have also been studying biomarkers for the different places of grapes cultivation. And now we are starting to also to do biomarkers for wine obtained from these different sources of grapes. So metabolomics has no limit for the imagination. We can find biomarkers in whatever we imagine. There may be some. We can also detect biomarkers for diseases, different metabolites in urine, for instance from people with certain diseases. We can also try to develop methods for detecting the origin of food. Rice, for instance, what is the difference between rice, Portuguese rices, several types of Portuguese rices? Is it important? Yes, for fraud detection. The price of one type of rice is not the same as another one. So we can also try to find biomarkers for water quality, art painting, whatever one can imagine. So for the moment, the next item to develop, and the facility has already started to develop it, is protein identification. We are trying to separate proteins by electrophoresis and then identify it using the BioISI mass facility. So this topic is just starting to be implemented. So this is the facility. We are also hoping to have new items to develop. Of course, all the studies have to be paid. We have special prices for the BioISI researchers. So, hoping to see you soon and that you have appreciated the BioISI Spec mass facility.
My name is Margarida Gama-Carvalho and I'm the co-coordinator of the BioISI Genomics facility along with my colleague Ricardo Dias. At BioISI Genomics, we offer an innovative certified multi-site platform that enables the on-site and real-time acquisition of integrated biological knowledge. Our vision is to contribute to the unveiling of the web of life, focusing on the digitization of biological systems, promoting the development of synthetic biology, and consolidating the concept of Biology 4.0. Our mission is to support national and international researchers and companies in the food, biotechnology, health, and agricultural industries in their transition towards a smart and sustainable economy, able to tackle the many pressing societal challenges of our time. Our core approach relies on a multi-source and multi-component single-molecule biosensing based on nanopore technology, supporting on-site real-time production of information and a fast-paced transition of data to knowledge. To accomplish this mission, BioISI Genomics is equipped with standard molecular biology equipments, including real-time PCR, a customized microfluidic platform for sample manipulation and processing, and Oxford nanopore equipments for real-time biosensing, with a current capacity to generate 1 to 2 terabytes of native DNA and RNA sequence data per day. As part of our data to knowledge concept, we further developed customized data analysis pipelines supported by a dedicated computational infrastructure featuring 8 CPUs, 9,520 GPUs, and 440 TPUs. This infrastructure allows us to provide a vast array of analysis from a variety of sample sources, reflecting on the many ongoing services and projects. A significant number of projects focus on the mapping of biodiversity, in particular related to the oceans, as exemplified by the recently funded MARCODE project. Other lines of service focus on plant biotechnology and promotion of smart agriculture, many of which in strong connection with the wine industry. Adding to this, we further support research projects in human health and epidemiology. The relevance of our activities have supported our integration into the Genome PT National Infrastructure for Genomics, first as observers and starting in 2020 as full members in National Reference Lab for Nanopore Technologies, further promoting our mission and values. BioISI Genomics, bringing knowledge to you anytime, anywhere.
Hello everyone. So I hope that you've enjoyed so far our facilities presentation, and despite the fact that we are a few minutes ahead of time, this is one of the advantages of having the system totally online. I apologize for 1 or 2 slight interruptions. It was just to make sure that you were paying the right attention to it. And if you have any questions or comments that you wish to make at this point, please do so. As you saw in the presentation, we also have, although with a slightly different input, we have what we designate as infrastructures, and members responsible for those infrastructures are also here. So if you want to ask any or make again any comment regarding that, the possible use of those infrastructures, please let us know. I added to the chat the names of the people which are responsible, and in our website you can find their email addresses and contacts. So in any situation you can contact and ask to book one of the equipments or to get the training done. Of course, we are enforcing these pandemic rules, and in that respect we have in this case now a more limited training for the young ones, young in terms of not being able to use the equipment on their own. The policy that we use is that first the person has to be trained in order to be independent in the operation of the equipment, and after that he or she will have simply to book the system through our Agenda platform, then use it, and in case there's a requirement for data analysis, that will be provided upon agreement with our staff, with the technical or researcher staff.
Sure that people will have very specific questions, so there is no need to put them now here, and you can always contact by email or through our webpage, you can see what services can be provided so that you can use them later. I think we are now in time for a small recess for coffee. I hope you have some coffee available near you, because we cannot provide it through the internet. And we will reconvene here at 11, maybe a few minutes before, so to make sure everyone has time to join. So see you in about half an hour at 11.
Thank you.
Good morning, everyone. I'm Manuela Pereira, and I have the pleasure to be the chair of this session on internal collaborative projects. And these projects aim at promoting excellence and novelty with a strong focus on interdisciplinary. So going across the different BioISI thematic lines in order to foster this integrative science that is one of the missions of BioISI. So I would like also to point out and make a special attention that the call for these internal projects for the next year of 2021 is now open, and please take advantage of this day to make brainstormings, to be inspired, get inspired, and write down your project. Especially if you are a young PI at BioISI, these projects are for you. So please do take the chance of it. And so, not with further ado, well, I want to present the first speaker of today's session, but just a parenthesis to mention that each speaker have 8 minutes and then 2 minutes of questions. So I would like also to challenge everyone to pose at least 2 to 3 questions. Please write down on the chat to me your name, and I will call your name to for you to ask the respective question. So the first speaker is Federico Herrera, and will talk about restoring NKX6 to function by protein complementation, so a proof of concept. Thank you, Fede. You can start.
All right. So my laboratory. Let me see if I can move this on. My laboratory is particularly interested in the fundamentals of neurodegeneration, and we originally try to go from the patients and the clinical features of neurodegenerative disorders towards the production of experimental models, either in living cells or in animals, and then try to test diverse, check the molecular mechanisms, and try to test different strategies for the treatment. Okay, but in the latest years, I'm focusing more and more on rare neurodegenerative disorders that are not very considered or very interesting for big pharma, but actually could make any knowledge on those disorders could make a difference for thousands or millions of patients. No, basically any neurodegenerative disorder that is not Parkinson's disease or Alzheimer's disease is rare, and many of them are caused by proteins for which we don't have a particular function. So from the academic point of view, they are very interesting subjects of research. Okay. So 2 examples I'm going to show here is one is an ataxia, a rare form of ataxia known as RSAX, caused by mutations in the saxin protein, and another one is Alexander disease, which is a neurodegenerative disorder usually of babies. That is very interesting because it involves the mutation of the GFAP protein, a protein of the cytoskeleton, which when it's mutated, it aggregates. Okay, you see pictures here. When you remove saxin from cells, the cytoskeleton falls apart, the cytoskeleton of the intermediate filaments, and we have very nice pictures obtained in the BioISI facility, imaging facility that Rui Malhó talked and Hugo Botelho talked before. Okay, here you see in the right, you see a normal GFAP distribution. It's a cytoskeletal protein. When it's mutated, it forms aggregates in cells, pathological aggregates. We published recently a paper where we were able to tag human GFAP, something that it was very difficult because the protein did not accept very well tags in the end of the C-terminus. So we, using a transposon strategy, put the tag in the middle of the protein, and we were successful, as you can see in the images. So this is 2 of the projects that we are working on right now, but I will talk to you a little bit more about the project that was funded by BioISI in a collaboration with Margarida Gama-Carvalho, Luísa, and possibly in the future with people in the bioinformatics section of the unit. No? In this project, what we are trying to work out is how to fix the function of a transcriptional repressor known as N-kappa C6 2, And this transcriptional repressor, when it's mutated, it forms a rare form of ataxia known as SPAX8. Okay, the mutations that are most common in this gene are nonsense mutations that induce stop codons in the middle of the protein, and therefore it produces truncated forms of messenger RNAs. These truncated forms can be degraded by nonsense-mediated mRNA decay, but the surviving mRNA molecules produce truncated proteins. No? So the idea would be to use a concept known as protein complementation, which is basically putting the remaining fragment of the protein in there and see if it restores the function of the protein, avoiding at the same time nonsense-mediated mRNA decay, okay, to produce more fragments of NK-6 and be able to induce the complementation with the fragments that are missing in the protein. Protein complementation is the second focus of our laboratory. No, rare neurodegenerative disorders. Protein complementation is the second because it's a universal property of proteins that allows several technical applications, like the one I'm going to show you in a minute, where any protein can be split into non-functional fragments that when are brought back together, reconstitute the function, and can be used, for example, for detection of protein-protein interactions or to kill specific cancer cells, etc., etc., etc. No? Here you see an example, is the green fluorescent protein, can be split in two non-fluorescent halves that reconstitute fluorescence when they are brought back together. No? And this is the basis of the molecular fluorescent complementation assay that we are using extensively in the lab, where we fuse two proteins of interest to two non-fluorescent halves of a fluorescent protein like Venus. No? And then when the two proteins of interest interact, then the fluorescence is reconstituted, the two split fragments of Venus are brought back together, and this fluorescence can be monitored and recorded by different methods like flow cytometry or microscopy. Okay, this is a very much used system in the lab, and we used in many papers in the past this system to see the interactions of proteins related to neurodegeneration that form aggregates in cells, like huntingtin or alpha-synuclein or tau in Alzheimer's disease. And we combine them between them and see how they co-aggregate, how they influence the aggregation of each other, and how they locate or where they locate in the cell. And this has been published repeatedly. Now I'm collaborating with Claudio Gomez at the BioISI to see the interaction between tau and S100B, okay, with a similar system by microscopy. Now, the third focus of our interest of our laboratory is the role of post-translational modifications on the behavior of protein dimers and oligomers. And we are using this system to change or to modify the proteins of interest in a way that they simulate or prevent a particular post-translational modifications. And we have published already for that two important papers, one by my previous PhD student, Joana Branco Santos, where we observed the role of phosphorylation on the aggregation and toxicity of mutant huntingtin for Huntington disease, and another by Ricardo Vilela and Beatriz Cardoso, where we show that asymmetric post-translational modifications could modify the behavior of STAT3 dimers, which is a transcription factor involved in neurodegeneration, but also in cancer and other disorders. Okay, so if I am lucky, I will be able to show you two videos that were done by Andrea Gutierrez at the BioISI imaging facility of cells where you will see at the left side cells with mutant huntingtin, which form aggregates, and then mutant huntingtin at the right where mutant huntingtin is phosphorylated, and you will see the difference in the behavior of the aggregates. In the left side, you see that the aggregates are more or less, they vibrate, but they are more or less stable. They don't change shape too much, no? And now you will see how the phosphorylated huntingtin behaves. It's more fluid. It's like liquid-liquid phase transition example, where the aggregates are more fluid and they exchange a lot of matter with the medium. They fuse, they separate, and they move more freely through the cell. Okay, this is an example of what we do in the lab right now. Live cell imaging, And now in the end, the cell forms an agrosome, another structure we are interested in, because the mechanisms are still unknown. And with this, I think I finish just thanking the bioimaging facility and the cell culture facility also at the BioISI, because they are essential for our work, the funding and the people who made possible these results. Andrea is here, the one for the Huntington project, Ricardo, the one for STAT3, Fernanda for our SACs, and Mika also for STAT3. Okay, thank you very much. I hope it was well because of the network issues and my mask. Thank you very much.
Thank you, Fed. So any questions? Well, as I said before, you can please do write your name in the chat so I can see who would like to ask some question if there is no question. So the next speaker is Bárbara Henriques, and Bárbara will talk to us about the multidisciplinary approach to study post-translational modifications in metabolic enzymes. Hi, Bárbara, you may start.
Hi, hello, everyone. No. Okay, I think everyone is seeing. So I will talk about my collaborative project with Bruno Vitor, and this involves two thematic lines, the biomedicine and biological chemistry. And we like to start by acknowledging our groups, especially Joana Ribeiro and Pedro Magalhães, that have been doing the hard work, and also Claudio Gomes for all the discussions and insights in the project. Let me just try to change the pointer. Okay, so this project has been dedicated to a special type of protein translational modifications, the acylations, and these modifications, we put an acyl group in the lysine side chains of proteins. And although, for instance, acylation is well known and is known for a long time ago, in the last years, new acylations are emerging. And it seems that, for instance, succinylation and glutyrylation are very important. And why is that? Because this can occur non-enzymatically. So imagine if you are in a state in the cell that there's accumulation of succinyl-CoA or glutaryl-CoA. These molecules can autocatalytically form this cyclic anhydride, and this is a very reactive species that will attack proteins and form these modifications on lysines. So in the last years, it has been shown that this acyl-CoA species can induce like a carbon stress very similar to the oxidative stress. Moreover, it has been shown that this aberrant protein acylation is a common observation in different inborn areas of metabolism. So this is known to occur, but how these modifications regulate enzyme function is not well established. So aiming to get some knowledge on these questions, we decided to study non-enzymatic PTMs, mainly succinylation and glutyrylation, on the electron transfer protein. This protein is involved in fatty acid, amino acid, and choline metabolism. That's why that's so important in the cell. And also, if you have mutations on ETF, you will get a disorder called multiple acyl-CoA dehydrogenase deficiency. So our project is on light on three different aims, and I will try to show you today some of the results we already have on these aims. So our first aim was to determine how these non-enzymatic PTMs could influence ETF function, structure, and stability. So first thing to do was to in vitro succinylate our protein. And just by incubating with this metabolite in alkaline conditions, as you can see here, using Resenborn methodologies, using an anti-succinyl lysine antibody, we see that we have modification a long time. And here, 10 minutes after incubation, we already see that some protein is modified. And this is at a larger extent when you get 1 hour. So we decide to analyze the two different conditions. And here you can see the function, how we evaluate the ETF function doing an enzymatic assay. And as you can see, the succinylated protein after the 10 minutes already has lost around 40% of the activity. When we have the condition of 1 hour, as you can see, the activity has diminished to around 10%. So for sure, when we succinylate ETF, it has a negative impact in enzyme function. So we decide to look at structure. We have done different spectroscopic methods, but here I will show you what we have using circular dichroism, and this will report on the secondary structure characteristics. And as you can see for the two spectra, we don't see major difference in the overall fold of the protein. So it seems that when we have the succinylated protein, the fold of the protein is not changed. But then we move forward and make some thermal denaturation studies. And here you have the control. And as you can see, there's a two-stage cooperative unfolding transition. But when we have the succinylated form, this transition is different. And we don't even get the post-transition part of the curve. So it seems that this succinylated protein is somewhat more stable, or maybe just makes like a rock. So this is still ongoing work, and we are still trying to dissect these questions. So then, at the same time we were in the lab doing this kind of experiments, Bruno and his group were trying to mimic similar experiments, but using computational methods. So for ETF, we have the structure, the crystallographic structure has been determined. And we also have a structure that you can see here that is ETF, okay, in blue, in complex with one of the partners. So this is medium acyl-CoA dehydrogenase. So the first thing to do was to parameterize this new amino acid, the succinylated glycines. So after doing that work and using homology modeling, they have constructed two new models where we have the ETF succinylated alone and also this ETF succinylated but in complex with MCAD. So we have 4 different systems, and then we have performed molecular dynamic studies. And here is shown RMSD graph. Okay, so we have 5 replicates for each system. And for each replicate, we have 2.5 microseconds FMD simulations. Here in the red line, it's the wild type alone with control, with no succinylations. And you can see here that you have more or less converged system. This is not the case for the other systems. So it seems that ETF, for instance, the yellow one succinylated, can sample in different, is still sampling in different conformations. So it's still diverging. And the same is true for the complex forms. So this means that we need more time of simulation and we need to do more replicates so we can for sure take conclusions from our systems. But still, and to show you some of the results we can take from this kind of studies, I will show you some of the analysis some of the preliminary analysis that we have already done. So here you have an histogram where you can see that for the wild type ETF seems to sample, like we have saw before the convergence, in one conformational space. And when we have the ETF succinylated, the conformational space, it samples in different conformational space. This is also true for the complex form where you see that it samples more conformational spaces. We can also look and analyze carbon alpha in the protein structure. And here is the case where you have the ETF alone, and we don't see major difference in the overfold of the ETF. Interesting, when we look at the ETF succinylated in the complex, and we'll make a difference with the normal ETF, we see we see some difference in the FAD region and in this region that it's called recognition loop that you can see here. And these are two very important regions for the enzyme function. Moreover, we can take other measure, and it's about FAD cofactor distance. So as you know, ETF has one cofactor, and MCAD, that is a homotetramer, it has 4 different cofactors. We can measure the distance between the FADs and make, for instance, a correlation graph where we, where we see the distance between the FAD and the other FAD on MCAD, and we can correlate. And what we see here is that in green we have the control protein, and in blue we have the succinylated. And as you can see, the ETF succinylated seems to sample different conformational spaces. Okay. Then we move to aim 3, and this was to validate some of relevant sites that could be succinylated. We decided to study 2 relevant sites, and these are these 2 lysines that flank the flavin cofactor. And for now, I'm going to just to show our preliminary results on this mutant. So we made 2 different mutants. One where we mutate the lysine for an arginine, so we would maintain the same type of amino acid. And in another mutant, we mutated the lysine for an glutamate. So we will introduce a negative charge that mimics the effect of the succinylation in that position. And as you can see here, when we evaluate the function of the enzyme, the arginine mutant doesn't change the activity, but when we put a negative charge in that position, the activity drops to 50%. So it means that that position will be very important for the function of the enzyme. So we had to succinylate these mutants, we could succinylate them, and now we are evaluating them. But for now, I can only show you the activity results we have. And for the arginine mutant, we see that when we succinylate the protein, there's still a drop in the activity. And when we compare, for instance, with the other mutant, the glutamate mutant, you see that it starts with a lower activity, like I already showed you. But then when we succinylate it, you see a higher drop. So this means that this lysine is important for the activity of the protein, and when succinylate will have an impact, but will not for sure be the only one to have an impact. So for summarize, we have be able to succinylate in vitro the ETF. We show that it impairs the function and the thermal stability of the protein. We have also in silico constructed models of succinylated ETF alone and also in complex with MCAT. We know that we need more time of simulation, and we have shown that the lysine 200 entity is important as an important role in enzyme function. Thank you all.
Thank you, Barbara. Any questions for Barbara at this point? So Barbara, maybe you mentioned it at the beginning and I didn't. Oh, there is one question. So Carlos Farinha, you may go ahead with your question.
Still trying to. Yes, Carlos.
I didn't make a question. I was just clapping to Barbara's presentation.
Oh, okay. Sorry, I saw your hands on. Okay, so I'll go with mine. Maybe if you mentioned it in the beginning and I didn't really pay much attention, but are these post-translational modifications, do they really occur concertantly or they are just random? Why did you do these succinylations?
So it has been reported in several papers, and people really believe in this, that these modifications occur when you have this accumulation of these metabolites, and they have been seen and identified. What we can't understand is what is the effect and what are the enzymes that are more prone to have these modifications. But what has been proposed is that all the enzymes that are involving in the pathways that lead to the accumulation of these metabolites will be more affected by these modifications. So imagine it's just a question of being there at the same time as the metabolites. So they are nearby, and as I show you, and this has been shown, the succinyl-CoA and the glutaryl-CoA are the most reactive ones, because they form this cyclic anhydride, and these species are very reactively. So for instance, for acetylation, you need to have these acetyl enzymes, to do the modification, but it seems that this succinylation and glutaryl-CoA can just occur.
Okay, do you think that they occur always at the same place, or they can occur at different places in the enzyme?
It seems to occur, in the enzyme or in different compartments in the cell?
In the enzyme.
In the enzyme.
In the same enzyme, like in ETF, like you were showing?
Well, I think they can just occur in any lysine that is available, but maybe...
So it's not specific.
Yeah, but maybe if you have some lysines that would have higher impact, they will, well, it is still to understand, and that's our question, if they have an impact on the regulation of these enzymes.
Okay, thank you, Barbara. So we'll have to move on, and the next speaker is Ina Uliyakina, and she will talk to us about exploring the impact of Staphylococcus aureus on cystic fibrosis, epithelial cell inflammation, differentiation, and epithelial repair. So Ina, you may start. Oh, I think Ines Panconian will also collaborate in this presentation. So you two may start. Thank you.
Can you see my screen?
Yes. Okay. At least we see a screen. I'm not sure if it's yours.
Let me know when it will be available, please.
Yeah, please do. Please continue.
Okay. So good morning. Thank you very much for giving this opportunity to present our work at BioISI Research Day today. And also we would like to thank, obviously, BioISI for providing us funding to perform this project. Our project is entitled Exploring the Impact of Staphylococcus aureus on Cystic Fibrosis, Epithelial Cell Inflammation, Differentiation, and Epithelial Repair. And as Manuela mentioned, both Ines from FunGP and myself from Bioenergetics Group will be presenting this work today. So I will start with a short background information regarding CF and S. aureus. So CF is caused by mutations in the CFTR gene, and as you may know, CFTR dysfunction leads to thick mucus that cause inefficient mucosal clearance that lead to recurrent bacterial infections and chronic inflammation. Cystic fibrosis individuals are predominantly infected by Staph aureus in the early age of their life, and later on they get also infected with Pseudomonas aeruginosa. Nevertheless, the roles of these two pathogens are not completely understood in the context of CF. So recently it has been published that S. aureus, that is a gram-positive bacteria, is able to release extracellular vesicles that we will be calling CEVs in this presentation, that are able to transport a variety of biologically active cargoes such as lipids, proteins, DNA, and RNA. And also CEVs have been shown to mediate pathophysiological functions by inducing cellular inflammation and even cellular death. So our hypothesis here is that CEVs may play a role also in host pathogen interaction in airway epithelial cells. So our main aim is to investigate the impact of CEVs during infection of CF airway epithelial cells with focus on inflammation, differentiation, and repair. So here I will show you a short overview of the workflow that we were optimization. So it corresponds to our first aim of the project, that was to produce and to optimize the process of production and purification of CEVs. So we have liquid cultures of Staph aureus, then we spin bacteria and continue working only with supernatants. And we filter supernatant. We then concentrate it using protein concentrators. So here we will be already restricting the size of the particles, like everything that is smaller than 100 kilodaltons is removed, and everything that is larger than 0.2 micrometers is also removed. So at the end, we will have concentrated supernatants that we can either inject into the column to remove straight away from the contaminants, or use also a commercially available precipitate reagent for the isolation of exosomes. And we will have our CEF formulation. Also, this formulation can be injected later on into the size exclusion chromatography column, and the spectra will look like we have vesicles, the first peak, and then secreted protein. Although when we do it for the CEF formulation precipitated with the reagent, the secreted protein fraction is not existent, so it's actually quite a pure formulation. So we wanted at the beginning to test different concentrations of vesicles, started from 0.1 micrograms per ml up to 100. And we can see that actually there is a reduced airway epithelial cells viability with increasing concentrations of CEFs, as you can also see from the pictures here. And this effect is more pronounced for the mutant CFTR stably expressing cells, as you can see from the green bars, rather than for the wild type. We wanted also to perform further dose response studies, and we used metabolic assays to calculate the redox potential of the cells. And we can see here that in green, increasing concentrations of vesicles will be reducing the redox potential of the cells, so reducing metabolic activity of the cells. And this effect was similar to wild type and F5-ODL CFTR expressing cells. Nevertheless, also the boiled vesicles had a delayed effect, as you can see, but still with higher concentrations, even boiled vesicles that we consider as destroyed vesicles showed some effect. Ines?
Okay, so hello everybody. I will continue now the presentation. So one of our first questions was then also to see whether these isolated vesicles have an effect on the expression of CFTR, the protein that is actually mutated in cystic fibrosis. And so what we are showing here is the Western blot. On the left-hand side is the result for the wild type cells. And what we actually saw is when we add vesicles to the wild type cells, we see an increase in the expression of CFTR. And on the right-hand side, we have the mutated cell line. And the same here, we actually see an increase when we treat the cells with vesicles. So this is pointing to a defense mechanism, which is then triggering or inducing like a mucociliary clearance. Yes, furthermore, next slide. Yeah. We also wanted to look into the direction of inflammation. So we checked the expression in those two cell lines of an intercellular adhesion molecule, which plays a role in the adhesion and migration of immune cells. But it has also been shown that it's the receptor for internalization of the rhinovirus. And so again, for the wild type cells, we actually see as well an increase in this expression of the ICAM molecule or protein. And for the mutated cell line, we also see an effect, although it's very small. And yes, so this is actually suggesting that these vesicles alone already trigger an immune response in the cells. And then, moreover, it has been shown already that Staphylococcus aureus, the bacteria itself, actually is internalized through a fibronectin integrin signaling pathway. And we wondered if these vesicles alone actually also somehow induce or trigger this pathway or this signaling. And so we used these cells, but we were growing them on filters, so they are polarized. And we checked for the fibronectin secretion in the supernatant. And as you can see, we actually found an increase in the supernatant in the wild-type cells, and the same in the mutated cell line. So yes, as a conclusion, we can also say that the vesicles alone somehow induce this signaling already. Another question was if these CEFs, these vesicles, have an effect on wound healing, which consists of two actually processes, proliferation and migration. And on the left-hand side, you can see the image. So in the wound healing, we actually monitor. So we have a cell monolayer. We perform a scratch, and then we monitor the wound healing over time. So as you can see, and in wild-type cells, we actually didn't see a huge difference. However, for the delta F cells, when we treated them with CEFs, we saw that the vesicles actually induced or accelerate the wound healing. And now we have to actually find out whether it's increasing the proliferation or migration, which will be a next step. Yes, so to summarize our project so far, we optimized the method for the isolation of extracellular vesicles from Staphylococcus aureus. We have seen that they reduce the cell viability and metabolic activity in human bronchoepithelial cells in a dose-dependent manner. And we also saw that the vesicles alone can increase the expression of CFTR in both cell lines, as well as the intercellular adhesion molecule. And they also trigger the fibronectin secretion to the supernatant. So our main conclusion now is that these as aureus secreted extracellular vesicles, so without any living bacteria, play an important role already in the host pathogen interaction or communication. Yes, and that's it. Thank you very much.
Thank you, Ina and Ines. So, any questions? I'd like to ask the help of the others, because Ina works in my lab and we have thoroughly discussed this project. So I think it's not.
I'm Carlos Farinha.
Okay, so go ahead, Carlos, please do.
So, do you plan? Congratulations on the work, Ina and Ines. Do you plan to test also live bacteria to check if the effects that you are getting from the purified extracellular vesicles is really mimicking what happens if you add live bacteria to the cells, to the wild type and F508del cells?
So obviously it can be a very interesting experiment, but I think currently we are just limited by the let's say work in accessible conditions. So Staph aureus, as you may know, is S2, and we actually don't have any incubator where we could co-incubate cells with living bacteria. But it's a very interesting suggestion, and obviously we would like to compare the effect.
Thanks.
Yeah. Thank you for the question.
Any other question? I've seen now in the chat, Peter Jordan wanted to ask a question. I'm not sure if it was to Ina and Ines or maybe before to
It was for the previous one, but I have another question.
Okay, great. So go ahead,
congratulations to the work, very nice. Do you have any indication whether the effects of the vesicles are due to docking to the cell surface of the cells, or whether the vesicles are actually incorporated into the cells and they are maybe release nucleic acids or any other components?
So I would say most likely they are actually internalized, because we know that the vesicles are getting internalized, and then they probably just release whatever protein, toxins, or DNA, RNA. Yeah, but this is something that we actually also want to find out which component actually are inducing all these signaling or these protein changes.
Okay.
Also, we would like to perform some imaging using microscope, like uptake studies, where we have bacteria and cells, at least bacteria loaded with a dye, and we can then follow their uptake during the live cell imaging. So I think this will also answer to the question. But generally for extracellular vesicles, you also have some docking and integration in the cell membrane. But for the bacterial vesicles, I'm actually not sure.
Okay, we have to move on, unfortunately. So thank you, Ina and Ines. So we'll move with Ana Catarina Alves. She will talk to us about the novel mechanisms causing familiar hypercholesteremia, functional characterization of variants into regulatory regions. So Catarina, you may start.
Okay, thank you. I now come. Just a moment. Okay, so thank you for the opportunity to present this work, and thank you for defending the project. The project is novel mechanism causing familial hypercholesterolemia, functional characterization of variants in the regulatory regions of PCSK9 and LDLR genes. I'm a PI and Julian Menezes are a co-PI. So familial hypercholesterolemia is an autosomic dominant disorder with high prevalence in most populations. It is characterized by increased levels of total and LDL cholesterol. The high LDL levels lead to accumulation of cholesterol in the peripheral tissues and accelerate atherosclerosis, thereby increased the risk of premature coronary heart disease. FH is a monogenic disorder caused by mutations in LDLR, ApoB, and PCSK9 genes. Mutations in LDLR genes are the most common cause of FH. However, about 50% of the clinical FH cases do not have a putative pathogenic mutations in the region study in these three genes. So the study of all genes by NGS and study of other disease mechanisms can increase the correct identification of these patients. Here is the LDL pathway. Briefly, the LDL receptor is a cell surface glycoprotein that is synthesized in the immature protein and proceeds in the Golgi apparatus, producing the mature form that is transported to the cell surface. There, the receptors binding to the LDL protein by apolipoprotein B. So now this complex is internalization by via claritine cockpit, and this complex is transported via early endosomes to late endosomal compartment. Here in the acid environment cause the dissociation of the receptor to the particle, and particle is recycled in the lysosome, is degraded in the lysosomes, and receptor is recycled to the cell surface again. Mutations in LDLR can affect different parts of the cycles and expression of LDLR. But recently, a new player has been found, the PCSK9 protein. When there is a gain of function mutation in PCSK9, the PCSK9 bindings to the receptor and this complex receptor in the LDL, and it is internalized. But here in the endosomen, by acid environment, this ligation is very strong, and it's not possible to dissociate. So all complex, including PCSK9, is degraded, and the recycle to the cell surface not occur. The variants in the coding regions of LDLR and PCSK9 genes are already studied in all labs. but the promoter and the 5-UTR regions is poor study. And so there are still few variant study. The aim of this project is to define the 5-UTR and promoter regions of the PCSK9 gene, as well as to perform an in vitro characterization of variants in LDLR and PCSK9 gene in the regulatory regions mentioned above. This project will also contribute to an accurate identification of FH patients, allowing for a better patients management. So our project is divided into 3 tasks. This is the first one. To study the impact of LDL promoter and the 5-UTR variants on LDLR, we cloned its promoter regions in the in-frame firefly luciferase coding by this plasmid. And obtain this, the LDLR wild-type PGL 4.10. Then, by site-direct mutagenesis, we obtained the constructs with the variants to study. Previous constructs, these previous constructs were expressed in the CHO cells for 24 hours and next to cell lines. Then the impact of the LDL promoter and the 5-UTR variants on the LDLR expressions was assessed by luminometry assays and RT-qPCR. The task 2 is divided in two. In the first is characterized the 5-UTR region of PCSK9 and next study the variants in the promoter and 5-UTR variants. To define the PCSK9 5-UTR sequencing, we use a 5-rise kit that involved the several steps described in this image. So the first results, we study 23 variants in the promoter region of the LDLR described in the ClinVar. ClinVar is a public and international platform where people report the variants they found. Usually put the information found the variants, but usually don't have information about clinical data or functional effect. So from these 23 variants, 56.5% result in a reduction of promoter strength compared to wild type. Most of these abnormal variants occur within in the sterol regulation element in repeat 3. It's an important regulatory element. We also study for variants in 5-UTR regions of LDLR, but none were shown to have a normal effect, just the negative control, which create a new ATG codon and has been demonstrated to produce an abnormal effect. So here is the first results for the 5-UTR region in PCSK9, and in the green bars indicate the annotated sequence for PCR 5-UTR, and the blue bars indicate the PCSK9 coding regions. And this red arrows is the indicate the indicate the 5-UTR identified by 5-rise for PCSK9 mRNA. So we pick the clones and sequencing, and when we sequence, we saw this same base. So this is a schema, and a schema to 5-UTR and promoter regions for LDLR, And this is 5-UTR and promoter regions for a PCSK9. This is a proposal. The conclusions, our results emphasize the necessity of functional analysis of new variants in the LDLR promoter with the objective of determining their biological effect and possible influence of FH phenotype, allowing the correct diagnosis. So during the pandemic situations, we only start the task 2 in August. So now we finish the task 2.1 and start the task 2.2, and in this moment are ongoing. And the task 3, the integration of functional results with the clinical and biochemical patient's data, is in this moment are currently collecting, because as I mentioned, some information is in the ClinVar, and ClinVar don't put the data about the clinical data about patients. So in this moment, we try to collect this information directly to the authors. Okay. And to end, thank you to all the research team to participate in this work and for the discussions. Thank you.
Thank you, Katerina. So are there any questions? Let me open the chat now so I can. So, Katerina, if I understood correctly, you would like to characterize all these genes, and you also think that they may have diagnostic potential. So you can use these for diagnosis. So how would you do that?
So the idea is increase the diagnosis, because 50% don't have a mutation in the coded region. So
okay.
The patient has a phenotype, but don't have a genotype. So our idea is exist variants in the non-coded regions, and need to study these regions to better identify these patients.
Okay, thank you very much. We are a bit late, but this just means that we are very enthusiastic about all these projects, and I would like to allow the speakers to speak, not as much as they want, but a little bit more. So our last speaker is Rita Pacheco, and Rita will talk to us about Val Health. So valorization of algae for health is a bioactive compound from marine bioresources by membrane technology. Rita has just pre-recorded her presentation, but Hugo, who is also involved in the project, he's here to answer our questions. So Rita can start. Thank you.
Good morning, everyone. My name is Rita Pacheco. I'm here to present Val Health, which is a project which I have in collaboration with Hugo Botelho from FundGP. The aim of our project is to identify novel bioactive compounds from seaweeds, edible seaweed. Those compounds should modulate not only cholesterol permeation in intestinal lining cells, but also the trafficking of protein CFTR, which is associated with cystic fibrosis, and the function of this protein in epithelial bronchial cells. We are also aware that various diseases are not well managed with the current available drugs, so it is mandatory to find new compound sources. We thought about edible seaweeds, as some diseases may be prevented by having healthy lifestyle habits. And in this case, an healthy diet is indicative of some compounds which may revert some of these diseases. In this case, we are talking about fucus, arame, and nori seaweeds, which are used in as edible seaweeds in cooking. So our targets are cardiovascular disease, which is the #1 cause of death globally. These patients have associated hypertension, diabetes, hyperlipidemia, high blood cholesterol levels, and also obesity, and they developed heart and blood vessels disorders, in this case the obstruction of arteries. Also, our target is cystic fibrosis, one of the most lethal genetic disorders in Caucasian population. It affects protein CFTR, and in the epithelial bronchial cells in the respiratory tract, accumulation of mucus is due to the malfunction of the transport of chloride ion. So in this case, a lot of infections in the respiratory tract and also in the epithelial tract of the body. In case of cardiovascular disease, we are trying to revert high blood cholesterol levels that occurs in this type of patients, or a risk factor of this type of diseases. In this case, the cholesterol level in the blood is a kind of a balance between of the biosynthesis of cholesterol in the liver, which is the protein HMG-CoA reductase, the main enzyme of the pathway, and also a balance of which we absorb in the food, in our diet. In the small intestine, we have proteins which are transporters of cholesterol into the bloodstream. In this case, when we are targeting to reverse high blood cholesterol levels, the drugs that are often used in the market are statins, which inhibit this enzyme, and also ezetimibe, which blocks the absorption in the intestinal lining. However, there are additional strategies to improve cardiovascular health, which is a healthy diet and also physical activity. In the case of cystic fibrosis, we are talking about malfunctioning of the protein CFTR. In this case, there are several types of mutations, but the most common one fails to transport this protein into the apical site of the cells in the respiratory tract, and the deficiency in the chloride channel blocks the mucus, turns the mucus very thick, and can lead to failure in the lung. In this case, in the market, there are several drugs which can be used. However, none of the current drugs can restore or fully restore the lung disease. So we know that CFTR is an ABC transporter, as well as other of the transporters in the intestinal lining cells, of the transporters of the cholesterol. They all belong to the same superfamily. Also in cystic fibrosis, there is an altered lipid metabolism, alterations in the absorption of the intestinal lipids. And we also are aware that diseases can be prevented by a healthy diet. So algae are known as presenting bioactive compounds that can promote or prevent several diseases. So the aim was to identify which of these compounds can have as target both of these diseases. So our task was first to isolate and identify these compounds from algae. We started by focus, and we developed a strategy to separate the different kinds of bioactive compounds present in the extracts. We are using membrane technology, and we are separating several types of fractions of compounds in fractions of the extract. Small size compounds, medium size compounds, large size compounds. We have seen that these extracts are rich in derivatives of fluoroglucinols, in this case fluorotannins, of different sizes, and we have separated them in different fractions in order to identify which ones were the most bioactive. We have characterize the extract and the fractions in terms of loose polysaccharide content, protein contents, fluoroglucinol contents, and also several activities. We are now in the progress of doing the same things for Arame and also for Nuri algae. We are also screening for this, for the activity of these bioactive compounds. In this case, for cardiovascular diseases, we are seeing inhibition of HNG-CoA reductase, inhibition of acetylcholinesterase, and also antioxidant activity. We are also studying cytotoxicities in cells which modulate the intestinal lining in the case of permeation of cholesterol, which are KECO2, and also cells that simulate respiratory tract cells, in this case CFBA. And we are doing viability assays in order to determine non-toxic concentration of the extracts that can be used to access or to screen the activity against the diseases. For the case of intestinal permeation of cholesterol, we are doing KECO2 monolayers, which are a model that simulate the intestinal lining, and we are studying the permeation of cholesterol in KECO2 cells in the presence of the algae bioactive compound. We are seeing if it can be blocked, the permeation of cholesterol with this type of compounds in order to decrease the intestinal absorption of cholesterol. And we are comparing with the current drug ezetimibe. For targeting fibrosis, cystic fibrosis, we are doing an high-throughput screening assay. In this case, we are seeing if the algae compounds can rescue the protein and putting the protein active in the apical side of the cell, transmembrane protein, and see if the compounds can have this activity. Also in progress, we have the studies of the effect of these compounds in the metabolomic profiles of the cells. In fact, the extracts are rich in all sorts of bioactive compounds. Some of these may have an effect in other targets of the cells. So we are extracting the metabolites from the cells which are in contact with the extracts, and then seeing or identifying these metabolites and comparing the cells in contact with the extract with the control cells and seeing if there are differences. In this case, the difference identified will lead us to the pathways that can be affected by the presence of the bioactive compounds of the extracts in the cells. We are doing this in the BioISI MS facility. So, due to COVID-19 restrictions, we have delayed the execution of the project. So by now, we can only conclude that seaweeds are rich in bioactive compounds, mostly fluorotides, which already demonstrated the potential to target cardiovascular diseases. We now have to go on and progress in the project until the March 31st and see if we can target also cystic fibrosis. We want to thank other team members, in this case in the participation of INZEL institution, Professor Miguel Mialma, which helped us in membrane technology, our master students who are working hardly in the project, and also all our volunteer students who are doing the permeations with Professor Miguel Mialma in the ISA. Thank you also for your attention. Thank you so much.
Okay, thank you. Okay, any questions? Hugo is here to answer questions that we may have. Okay. Okay, so I have a short one, I would say. So you are trying to characterize all these compounds from algae and see the effect of the different compounds. But can you also consider that you just don't have any effect of a single complex, but like a synergetic effect of several compounds, and that's what makes algae unique.
There is in fact most likely the effect, because as you noticed, we are not studying for pure compounds. We are studying more or less semi-pure fractions, and the focus of this project is not to isolate compounds. So we will not be able to distinguish whether the phenotypes we see are due to a single compound or to compound combinations. But that's one of the goals of this BioISI project is to provide so promising sources of compounds for future more detailed studies, looking at individual compounds and looking at their functions.
Okay, thank you, Hugo. So we have to finish, or a little bit behind schedule, so 8 minutes is not that much for this exciting discussion. I really enjoyed it, and I hope that you all have that you all got enthusiasm, and all the new PIs will rush and write down their application for the next call, which will be ending on the 15th January next year. So you still have some time, but please start writing it down. It's really funny, and it's good really to share our knowledge, our different perspectives, and have all this integration that really BioISI aims at. So thank you. I think this is done for the morning sessions. I don't know if anyone else has any thing to say. So let's go for lunch and come back at 2. See you. Bye.
Hello. Welcome all. I hope your lunch break was okay. And in order to keep our schedule, we're going to start the afternoon session, and the first one that we're going to hold is related to bioindustry. I think one of the things that this pandemic has shown is the importance of having science and society close together, and at BioISI we are proud that we are being able to do that. You've seen that as, for example, our fight against COVID and the input of our genomics facility. And here you will see other projects that intersect with different thematic lines, bioinformatics, biotechnology, biomedicine, and that show us that the gap between fundamental research and applied research is being narrowed, not only by the advances in technology, but also because of the willingness of our members to promote that narrowing of the gap. So we will start with a presentation by my colleague Anabela Bernardo da Silva, which will show us 2 very much related projects, Goji Berries and Optimal, that focus on the partnership between fruit industry and our plant functional genomics group. This first presentation was recorded, and it will start soon, and then the other ones will follow. As in the previous one, if you have any questions to the participants and the speakers, you can put them directly on the chat, and we will be dealing them as they arise. So once more, thank you very much for your presence, and we will start now the session with the Goji Berries and Optimal presentation.
Good afternoon. Today I will talk about Goji Berry and Optimal. They are 2 projects involved in fruit production. These projects are financed by PDR 2020, and they have specific conditions. All the projects must have at least 1 research institution, an agriculture, and also an association of agricultors. In these two projects, the school teams involve BioISI members, as you can see here, and also members by other research centers. In the goji berry project, the coordination belongs to Consulae. The research institution is Faculty of Science. This is the association involved, and also the agriculture is Monte das Bagas and organic production. The objective of goji berry is the production of goji plants, because you want to obtain not only the berries but also the leaf that can be important by consumption because they have a very high antioxidant capacity. And we want also to improve the production of berry and leaves in order that we can consume in fresh and after dehydration, and we want to understand what are the difference in these two conditions. We are using microbial inoculum to promote plant growth. We use different techniques to detect early change in the potential productivity of the culture, and we make also analysis of the nutrient in soil and plant in order to see if this is improved by the microbial inoculation, and of course the quantification of nutritional components of goji berries and leaves in fresh and after dehydration. The orchards that we are studying are near Lagoa de Santo André, Melides. They have 4 different sectors. 2 of them have the plants of Lycium chinense. This is the species with the greatest relevance in Portugal, and the other is the Lycium barbaro, as far as we know, the species that can produce the best goji berries. You must have in mind that when we talk about goji berry, we talk about berries that can be produced for more than 100 species, different species. So it's a very big confusion about it. Here we see also some kind of equipment that we use for the measurements. One of the equipments is the SPA. The SPA is a equipment that can estimate the soil and plant development. It can analyze the soil and plant development, given an index that can be related to the nitrogen in soil, nitrogen in plant, and also chlorophyll in plant because it's related with nitrogen. But in fact, in goji berry, in this case in Lycium chinense, we found no correlation between the SPA index and the chlorophyll content that we determine after extraction of the pigments and quantification by spectrophotometric techniques. So it cannot be used as a proxy to chlorophyll in goji berries. But we measure a lot of several different parameters, and we found one that is also used to study the performance, the overall photochemical performance of the leaves, the PI instantaneous, and these parameters shows a better performance, photochemical performance in leaves of the plants that are inoculated at root levels with the microbial, in this case a fungi. And we can understand, and so we can say to the agricultor, we have a parameter that can follow the performance and the behavior of the culture, and we can also make a deeper analysis of photochemical process involved, because we can study the other parameters, for example, the electron transport, so we can give us the information at what level the photochemical reaction in the leaves are affected by the inoculation at root level. In the optimal project, more entities are involved. The coordination belongs to the INIAVA Alcobaça and to Miguel Leão. We have 2 associations involved, 2 research institutions, and also 6 different societies of agriculture. The objective of this project, and this wider reason of this name, is the optimization of Maçã da Alcobaça. So the optimization of light intersection by the trees. Why it is important to study that? Because if you want to increase the yield, the production of an orchard, you think, okay, if I have more trees, I can have more production. But in fact, at this moment in the orchards, and some orchards in Alcobaça, you have so many trees per hectare, so many trees per line, as you can see here, this line, and the space between the lines can be so small that part of the light are intersected for only part of the leaves of the canopy, and the other leaves have a limitation of irradiance. So here you can see in this image Miguel Leão determining the intersection and the distribution of light radiation in trees, and also in these trees we make a detailed study of photosynthesis using the equipments similar to the one we use in goji berry, and also infrared gas analyzer techniques, and we of course study orchard production and fruit quality. So these orchards, today I will show results related to 4 different orchards, and these are all the orchards studied belongs to the region of the Alcobaça. This is a region with geographic localization that is protected, so that means it is important for the market. And we have here the trees, and we have here an example of the position of leaves where we measure photosynthesis in the trees. So for example, we can have a leaf oriented to east outside the canopy, and the other oriented to east but inside the canopy. And here we have, for example, the measurement of fluorescence of chlorophyll using a PIA. So again, looking for the spot index, in the 4 orchards with different densities, you can see we have 2 orchards with similar density, both with high density, one with average density, and the other one with very high density. But the orientation of the lines of the apple trees inside the orchard can be different. In this case, you have the same orientation, and we can compare one orchard with average density and the other with high density. And here you have 2 orchards with high density, and we can compare opposite orientation of the lines of the trees in the orchard. What is important to notice is that only in August that the plants are having the maximum photosynthesis, we found significant difference between spot index depending on the position of the leaf in the canopy. And this is depending of the intensity of the density of the orchard, and also orientation of the orchard. So we will have a lot of things to study. Of course, we test if the SPI index gives a good correlation with chlorophyll content. In this case, we have a very good correlation, so we can use SPI index as a proxy of chlorophyll content. But we can also optimize the intersection of light by the canopy, changing the pruning of the trees. And here we have 3 different examples. And for example, in other orchards that we have the three different pruning conditions, and when we measure fatty acids in apples, we can see that the fatty acid, the composition in fatty acid is affected by the pruning of the in two different orchards in the fatty in similar way, and also if you consider not only the pruning but also if the apples are obtained for inside the canopy or with less irradiance or outside the canopy, you have a stronger correlation with pruning and the position of the tree inside the canopy. So these are only some examples of the results we obtain in this kind of project. And here are also some examples of the different output we can have, because you can have outputs in international congress or in a magazine for agricultors. Thank you for your attention.
Okay, thank you very much, Manuela. You can now take questions. I would like just to make a short comment. One of our partnerships that Margarida Amaral mentioned in the beginning was our involvement in a collaborative laboratory, which has to deal with smart farming, and some of the tools and measurements that Lucia mentioned here can actually be applied to that. I know the fatty acids is still an invasive measurement, but things like the oxygens and respiratory changes and that sort of stuff can eventually in the very near future be measured with optical devices coupled to drones that can be operated by the farmer at a distance and then being analyzed remotely also. So this is one of the examples that we would like to promote, and I thanks very much, Annabella, for showing this to us this afternoon. Okay.
Thank you, Rui. And I can say that we also test several of the parameters that are measured by remote sensing. We determine now at this moment at leaf level in order to see, can decide what kind of parameter can be more promising to study all the productivity. Okay. Thank you.
Flavio just put a question on the chat that it was something that I was going to launch in the discussion later on, that would be a general question for the presenters of this session, and that is just how much, in this case farmers, but medical doctors or whatever our industry partners are now receptive to this kind of innovative measurements and techniques.
In fact, they are, most of them, they use SPAD. So is why I showed the results we obtained by SPAD. And I beginning also to use measurement of chlorophyll fluorescence, why I show also the results obtained with PIA. For example, the PI instantaneous. So there are all parameters that most of the agricultors are using. Sometimes what they ask us is to understand why they are measured that kind of difference, because they know that when they have difference, they must do another adubation or something like that, but they want to understand why. I think we need to have in mind that at this moment, most of them are agronomic engineers, so they study a lot. And okay. All of them are young and use all the new device easily.
Okay, thank you very much, Lucia. So I say Lucia because it's the name that we also familiarly treat Annabella. We will now proceed with Nuno Bandeira and on a project more focused on bioinformatics, and that will be computational modeling for improved photoactivated detergent microcapsules. Thank you very much, Nuno. Whenever you want, you can start and share your screen with us. Thank you.
Okay, can you hear me okay?
Yes.
Okay then. Right, so So I'd like to thank everybody for, especially the organizers, for inviting me and giving me the opportunity to talk about my association with an industrial partner. So I'm going to talk a little bit informally about how my partnership began, going all the way back to 2015. So back then I had a very generous European grant that allowed me to travel. And I had a friend of mine who was working at the world headquarters of Procter & Gamble, and he invited me over for, to give a small talk on site. So I went over to the US, and in case you're wondering, this is me looking like an idiot. So I gave a small talk, and I learned a little bit of what they were doing there and what their interest was. They have their own modeling center, so their main focus was on microcapsules. And you can see the microcapsules industry is pretty diverse. So P&G is mostly focused on household care products, and their research was basically focused on how microcapsules can be mechanically changed. The morphology can be mechanically changed through interaction with visible light. Now, one of the building blocks that they can use to do that is already been out there for quite some time, and it's been known extensively to be photosensitive. So you have azobenzene moieties, so azobenzene products which through the interaction with visible light can change or interchange between two different isomers. And these can be used as building blocks for the microcapsule walls. So you can see that there's a morphological change when you go over from the trans isomer to the cis isomer. And this allows for a sustained release of the active ingredient that is inside the microcapsule, the capsule shell. So the concept is you shine visible light on the capsule, and it kind of shrivels and allows for the active ingredient to be released in a sustained fashion, okay? Now, fast forward to 2018, I was already beginning my fellowship at BioISI, and I get a phone call from this friend of mine who had invited me over, and he asked me if I wanted to collaborate with them on a project, a specific project that they had in mind, specifically concerning the makeup of these microcapsules. So I had to sign a confidentiality agreement and a non-disclosure agreement, and I was involved in this project. And so essentially the work was essentially being done by a PhD student who was looking into these two monomers that could potentially be used for the microcapsules. So they screened through lots of products, and these two were target candidates. And you can see the effectiveness of the isomer conversion through absorption spectroscopy. So the bands here change as a function of time. So if you have signature bands belonging to one of the isomers, you can monitor them and see how they change. So with ASO1, you get one particular feature, which is it's very slow over time. So the way that it changes or converts to the cis isomer is a matter of hours, but with ASO2, it's a matter of minutes, as you can see here. So my main contribution, or the goal of the project, was to try to explain these features and look into the photochemical properties of these two azo-aryl molecules and see how they change. So there are lots of ways of modeling. This is just a sketch of the things that you can do. So if you start from first principles, you have what is called a molecular scale, and you can go all the way up to unit and process engineering when you do plant modeling and things like that, okay? So I'm going to be focusing on this part over here. So it's the one that concerns bond breaking, electronic phenomena, and you can access all kinds of properties by solving Schrodinger's equation. So these are the basic fundamental principles of quantum mechanics. So if you look into the two azo molecules that I've shown you before, I can summarily sketch the absorption spectrum features of these molecules right here. So you have to look into the makeup or the electronic structure of the molecules in the frontier orbital region. And you can see here that you have the nitrogen lone pairs. Okay, so you have also the pi bond, which is being formed between the two nitrogens, and you have the complementary pi star bond. And what happens is you get two types of bands. So you get an N to pi star band, which is weak and is usually accumulated in a bigger band, which is the pi to pi star band, which is relates to this transition, electronic transition from this one to this one. Now, the good thing about molecular quantum mechanics is that you can map out all the energies of all the manifold states that you can have with absorption spectrum, especially when you focus visible light onto the molecule. And if you follow the reaction coordinate, and in this case, you have to distort the dihedral angle between the plane of the rings and the double bond between the nitrogens, all the way from the trans isomer to the cis isomer. So you have to travel these angles right here, and you can map the energies of each of the states. So S0 is the fundamental, is the ground state, okay? And this is the first excited state. And this is the pi star state that I've mentioned before. So when you promote the electron to this state over here, you still have to allow for a certain thermal activation for the process to occur. So here we're talking about something that is not very usual or very common in chemistry, which is an excited state transition state. So you have to promote the electron onto this surface right here, and then you still have to overcome this energy barrier in order for the molecule to relax. And then when you come to this stage right here, you have what is known as an avoided crossing region. This allows the electron to tunnel through from here, this point, over to here, okay? And then you go all the way to the cis isomer. And then you monitor this through absorption spectroscopy again, because if you have a certain amount of cis molecules and not so many of trans molecules, you can show up in this can show up in the differential spectrum, okay? So here again are the two fundamental orbitals, okay? And we know that the rate of interconversion between the two isomers is going to be dependent on this energy of activation, which is something in the region of 36 kilojoules per mole. Now, all right, so now we know what happens in ASO1. This is consistent with experiment. Let's see what happens in ASO2. Now with ASO2, you get essentially pretty much qualitatively the same features, but here there's a small difference. So here you have an activation barrier of 9 kilojoules per mole less than the one in ASO1. So this, again, this is consistent with experimental findings. So the ASO1 converts much faster than ASO1. Sorry, ASO2 converts much faster than ASO1. So here, this allows us to conclude a few points. So you have essentially a slower rate of interconversion in ASO1. One hypothesis that was somehow presented was the possibility that somehow there would be a hydrogen bond between this hydroxy group over here and the two nitrogens that would hinder the rotation of the molecule. This didn't turn out to be the case, because if you look into the pi bond orders, and this can be easily achieved by analyzing the wave function of the excited state. Now, if you look into the pi bond orders, you formally, when you have this excited state, you should have a bond order that is close to 0. Okay, but there are some residual bond orders in this case, because the wave function is somehow polarized. So there are slight differences between these two values, which explain why the rotation takes place much faster in azo2. So with the two methoxy groups, methoxy groups over here, or the methyl groups over here and over here, you have kind of an injection of electron density that somehow releases or depolarizes the pi bond and allows the isomer to rotate into the cis form more easily than otherwise. So in case you want to know more, you can read all about it in our paper we published this year. And this is a photo of our collaborators, and I'd like to thank the funding sources for this, Procter & Gamble and these institutions, and my colleagues. And this is Domenico, who conducted all the experiments, or nearly all the experiments. And I'd like to thank you for your attention. And I'm open to any questions now.
Thank you, Nuno. Thanks very much. Again, here I have a small minor initial comment to do, is that for those that may not be so familiar with this, for example, we use analogs of these molecules that Nuno is trying to do. We call them like cage probes, where we have inactivated molecules of interest inside the cells that can modulate ion content, growth regulators, can be used to follow embryo development. So the interest of this molecule is quite substantial, especially in the biological sciences. So thank you a lot, Nuno. I don't know if anyone has any immediate question for him, or I can put it in the chat as well. Okay. So if not, this is this awkward seconds of silence, like when we're doing our lectures, where we ask the students if anyone has any questions, and we are learning how to deal with those seconds of silence. So we will move on to the next presentation. You have it in the program that was going to be given by Raquel Chaves, which is the responsible of Pulmagene. She unfortunately has a familiar situation that has to be dealt with regarding parent. And so the presentation will be made by the PhD involved in the project, Daniela Ferreira. Right? So Daniela, welcome, and you already start sharing the screen, so whenever you want, you can go. Thank you.
So good afternoon, and thank you for this invitation addressed to the PI of Pulmagene project, Raquel Chaves, to perform the presentation in the BioISI Research Day. My name is Daniela Ferreira, and I am a postdoc researcher of Pulmagene. I will present this project that is a collaboration with Stapvida and that aims to create a genetic blood test for cancer therapies. Lung cancer is the most common type of cancer worldwide, and non-small cell lung cancer represents about 80% of them. Our target in Pulmogene are these patients with an advanced stage of disease and who can really benefit from targeted therapies with tyrosine kinase inhibitors as afitinib that is currently used for EGFR mutated non-small cell lung cancer patients. Currently, PCR and Sanger sequencing techniques are used in order to detect specific mutations in tumor biopsies that are usually formalin fixed and paraffin imbibited. These alterations in specific genes as EGFR or ALK are considered as predictive biomarkers once they can determine the sensitivity or resistance to the treatment with a tyrosine kinase inhibitor, helping in choosing the targeted therapy. However, frequently some problems can be faced with solid biopsies as the DNA degradation in formalin fixed and paraffin imbibited tissues, the fact of biopsy method being too invasive, which limits the sample collection during the cancer treatment, the limitations in the amount of tissue, among others. So Pulmogene aims to help these patients in their response to therapy with a non-invasive product and with high sensitivity in detect mutations of interest. The goal of Pulmogene is create a new system that consists in a range of innovative reagents that allow cell-free DNA extraction from a simple blood collection, that is a liquid biopsy, reagents for the amplification of the regions with genetic alterations and its purification, and thus the detection of the specific mutations will be performed by next generation sequencing or high resolution melting or a biosensor that is developed in UTAD. This product will also include a simple software for automatic data analysis and issuing a medical report. This approach will allow a non-invasive follow-up of patients with non-small cell lung cancer during therapy, and this is very important as a high percentage of patients acquire specific mutations during treatment, and this drives to its resistance. So to develop the Pulmogene kit, we need to determine the best method of cell-free DNA preservation and extraction, define genes and mutations to be analyzed, design the experiment for the amplification of interest regions, and validate these results with liquid biopsies of patients with non-small cell lung cancer with and that need to also be compared with the results from solid biopsies. We started our research by determining the best method for cell-free DNA preservation and extraction in plasma samples from healthy donors. We test two different tubes, and the cell-free DNA collection tubes from Roche were the best for preservation of cell-free DNA. Also, they are approved for diagnostic use. The extraction of cell-free DNA 6 days after blood collection with these tubes revealed to be also successful. And the method for the cell-free DNA extraction based on magnetic beads with the GeneLut kit is the one with high recovery of cell-free DNA from plasma. The next step was define the genes and mutations to be analyzed and design the experiment for amplification of interest regions. As EGFR is one of the most frequently mutated genes in non-small cell lung cancer and is a predictive biomarker with a high number of target therapies, we start to develop the Pulmogene product with the analysis and mapping of EGFR mutations reported in COSMIC database that are predictive of treatment outcome in non-small cell lung cancer. These mutations are located at tyrosine kinase codifying exons 18, 19, 20, and 21, and are reported to be deletions, insertions, or point mutations. This analysis is essential for the design of a set of primers capable of amplifying these regions and include
all the described mutations in the multiplex PCR. After that, multiplex PCR was optimized for amplification of cell-free DNA samples, and the products purified with ExoSAP and magnetic beads methods, and then sequenced by next generation sequencing. All the EGFR exons amplified by multiplex PCR were identified by NGS, and furthermore, a specific deletion in EGFR exon 19 was detected with a frequency of 56%, demonstrated the value of this method in the EGFR mutation identification using cell-free DNA. The outcome of this deletion is sensitivity to the first and second generation of EGFR tyrosine kinase inhibitors. Also, we confirmed the result by Sanger sequencing, confirming that this is an heterozygous mutation. We developed the high-resolution melting analysis for EGFR exon 19 in order to have an alternative for NGS, and this method revealed to be highly sensitive, being possible to identify the mutation when it's present with an allele frequency of 0.25%. Also, this method revealed to be efficient for the detection of the previously reported mutation in cell-free DNA sample as an heterozygous variant. Regarding the future perspectives of this project, we aim to validate this method in liquid biopsies of patients with non-small cell lung cancer and compare with results from solid biopsies. We have established a collaboration with Hospital Sant Antonio to collect these samples, but we didn't start yet due to the current pandemic. Also, we aim to design experiments for analysis of other predictive biomarkers as ALK-gene, develop the high-resolution melting experiments for analysis of other EGFR exons and other genes, and test biosensor for analysis of EGFR mutations. Thank you for your attention.
Okay, thank you very much, Daniela. I just forgot to mention something that is relevant. Daniela and Raquel are in our poll from University of Trás-os-Montes e Altor in the north. So from a distance, hello, thank you very much for your contribution. Any questions to Daniela, the immediate ones? Okay. So we will now proceed with another contribution from UTAD, in this case, Paula Martins Lopes, and which will talk a bit about the related projects on wine and vine. One is actually called InnoVine and Wine and Interact, a partnership for the wine sector. Paula was yesterday awarded for the second time with the CNOIV Award. So from here from Lisbon, congratulations once more for this very interesting award. And Paula, whenever you want to start, please do so.
Thank you. Thank you for the invitation to present these two projects that are projects from the university where I belong to, so from the University of Trás-os-Montes and Alto Douro. But however, they had scientists from belonging to the BioISI teams that actually participated in them. And they are a strong way of seeing how research can be joined with the different sectors, production sectors, and here in particular the wine sector. These are two structural projects that we have participated. They belong, as I said, to the University of Trás-os-Montes, that was and Alto Douro, that were the promoters. There were Interact and InnoVine and Wine. The Interact project was divided in three different lines: Isaac, Best, and Vitality Wine. the people, the researchers from the BioISI Center participated both in exact and vitality wine. Whereas in the Innov wine, we had 4 different, and they are already, this one is directed to the wine sector. And here there were 4 lines of research, one dedicated to edaphic climatic conditions, the other one to viticulture, winemaking, and competitiveness, sorry. We participated, the BioISI team from UTAD participated both in viticulture and winemaking research lines. The aims of these 2 projects are really related with the sectors where we are integrated more directly to them. The Interact was trying to create opportunities with local economy, and most of them involving the agri-food sector. But they involved also the agri-food, not considering only the crops, but also considering the local breeds that we have here. Whereas in the Innov Vine and Wine, it was it started to as a foundation actually for now a collaborative lab that is now actually working, and this was the basis for this start of this collaborative lab. It was to promote outstanding research and knowledge, to transfer this at the national and at the international level. And this included all the wine chain sector, and it also included problems that are associated with environmental problems that can arise from the practices that we can find in the wineries and also at the vineyard level. So where did the group from the Plant Functional Genomic Group participated in the Interact Vitality Wine was more related with the problems that are related with heat stress. As we know, global warming is happening, and here the wine sector is really affected by this global warming. And so the group from UTAD was involved in studying the effects, the genetic effects that they had both in the plant per se, and also a result of the treatments that we can actually drive to the vineyards with the sun blockers, and how that affects both the plant and also the quality of production. In relation to the Innov Vine and Wine, they also participated in the viticulture line of research that was overlapping in some aspects with this Interact project. So I won't be speaking more about that. I will, in the Innov Vine and Wine, I will speak about one that I was directly involved in, the winemaking. That was one of the lines, the research lines that was related with wine authenticity. This wine authenticity research line also had the participation of another BioISI team, that is the microbiology and biotechnology group, through Alexandra Mendes Ferreira, that participated in this work with us. And when we look at the authenticity of wines, we have to look at 2 dimensions. We have to understand that we have one dimension has to be related with the geographical origin of the wine that is associated with the denomination of origin or appellation given to these wines, and the other one is related with the varietal composition. So in this line of research, we aimed to target both of these 2 dimensions in order to try to get a model that would work as a whole to detect the different compositions of the wine relating with varietal origin and with the geographical origin. And my group was more dedicated with the DNA aspect of the detection of the varietal origin that is present in each wine. So as we look into a label, a wine label, we see that they are composed of different grapevine varieties. Although there are several different ways of trying to detect the varietal origin, the one that is not affected throughout all the wine processing production is actually the DNA. The DNA that we find that is present in the grapevine, that is present in the grape, will be persistent and present in the wine. So my group has worked for already almost a decade in this problematic with the wine sector and with different wine companies. And we have developed a DNA-based system that is one that is based on PCR detection, and the other one that is avoiding PCR. Once PCR has a big problem, that is the inhibitors can actually avoid the amplification, and we can have problems in detecting it. So the first one that we actually aimed was try a PCR-based methodology that we detect the end product, that is using a high-resolution melting PCR, PCR. So what we designed this and what we developed this method was using wines that were produced by us, and that we followed them from the field to the bottle. So we controlled the entire process, and we knew exactly which variety was within each wine. So these are our standard references for all the different samples that we can actually test afterwards. And we put this method working, and here you can see one of the results that we got a high-resolution melting assay is able to detect different grapevine varieties in the wine, and it works, and it has exactly the same profile that we find in the leaf profiles. So here we are actually distinguishing 8 different profiles. 2 of them, if we see here, Touriga Nacional and Touriga Franca, actually have exactly the same profile. Because within this sequence that we were analyzing, they had the same exactly sequence. So we aren't able to distinguish with this assay. But this assay actually gives us an advantage over other assays that can be used PCR-based, like microsatellites, because we actually can detect differences that are present in very small regions, and actually can distinguish and are very well conserved between within the different grapevine varieties. We then also, using this information that we were gathering in terms of the differences between the grapevine varieties, we are targeted to find smaller samples, because one of the big problems when we're dealing with wine is actually that we have DNA that is highly degraded. And this highly degraded DNA might not work with PCR primers if we don't target very small samples. Very small sequences. So we started to develop biosensors that can be DNA based. We use small probes that actually can differentiate between different sequences between different grapevine varieties. So here is an example, and this one was actually the paper that won yesterday's award, where we used we used a sequence that was specific from Tinta Amarela, and that only is differentiated by Tinta Roriz in a unique SNP that is underlined in the Tinta Roriz sequence. And when we applied this to an optical biosensor, we actually could see that the sequence would only hybridize and will give us a positive shift of signal that is related with Tinta Amarela, as we see here. And this differentiation in terms of wavelength is significantly different from the non-target complementary target and also with the Tinta Roriz. So we demonstrated in this work that we are able to detect this small difference in only a position, a nucleotide position. And this is very interesting for the sector because we can actually use very small sequences and have a very high resolution using samples that aren't as purified as we have to have them when we're using PCR-based technology. This is one of the targets that we have to have is within the wine authenticities sector. Problematic is actually detecting the grapevine varietal composition, but the other dimension is detecting the origin. And here we had a collaboration with a group that is here in UTAD that works in geology, and they studied different compositions of the rock-based and of different elements, isotope elements, that can actually give a fingerprint of the origin. So based on these fingerprints, and here's one that shows us an example of how this works. We have here samples provenient from Alvarinho, that is one of the grapevine varieties that is used most in the north of Portugal, that has denominations associated to the Monção and Melgaço region. And we can actually differentiate this Alvarinho from that is produced, that here is in blue and in violet. We can differentiate it purple from the Alvarinho that is produced in the Douro region, that is in Bordeaux reddish. So here you can actually see that we can get a different profile that is related with the geographical origin, that is related with the bedrock where the different wines are produced. So here we have a system when we're designing this model that we can actually target all the dimensions of the geographical origin. So in order to understand how these two projects that belong to TAD as the main have contributed to the outputs of the scientific publications. Here we see how it has evolved throughout the years. So the project started in 2017, and we have produced and we increased the publications related to these. So thank you very much for your attention.
Okay, thank you, Paula. I think it's also part of my task to enhance our deeds, good deeds. And you didn't mention, but I know that this same technology you are applying in a project against COVID. So if you want to just to give not more than 1 minute, please, but if you want just to talk about that for a minute, I think that will be very interesting.
Yeah, actually, the biosensor that we started to develop was this one based on optical system, but now we are using a new one that is actually patent that presented there. That was a result of those of this project in Vine and Wine. And this basis of research, when the COVID outbreak started, we thought, okay, we have here a system that might work in COVID. So we started to work in it because it's all DNA, RNA based. They are all molecules, all the same throughout the different organisms. And actually we got that to work, and we had a project, and now we got actually recognized by the foundation of Mota, Antonio Manuel da Mota, that gave us an honorary prize on this research, because we're trying to get this in the market and trying to get a quick response, have a response of a positive or a negative presence of the SARS-CoV-2 presence in an individual in 20 minutes. We're able to get this working and get a result, a clear result, without having to extract RNA or anything. It's directly. So we get the swab, and after 20 minutes, we know if a person is positive or negative. So at the time being, we're just trying to get this now trying to legalize it and trying to get it approved by the institutions in order to spread it and to be able to be available in the market within, I hope, within 2-3 months. So.
Okay, great. Thank you very much, Paula. I can't give you more time. You have a question on the chat for you, so you could reply there, please. But we move on with Iris Silva, and she will present the last project of this session, titled Personalized Treatment for Cystic Fibrosis Patients. Iris, thank you so much, and whenever you want to start, please. We are not listening.
Hello? Yeah, now it is.
Okay, now yes.
So hello everyone. So thank you for this opportunity to show you our work in this European funded project, which is entitled HITCF, and its main aim is actually to find a personalized treatment for patients with cystic fibrosis that have ultra rare mutations. So as you, let me just take this from here. Never mind. So as you may heard from previous talks, cystic fibrosis is one of the most common genetic disorders in Caucasians. And it is caused by mutations in the CFTR gene, which is a chloride and bicarbonate channel located in the apical membrane of epithelial cells. And these patients also are often show progressive loss of lung function, which is the main cause of morbidity and mortality. And they also present elevated concentrations of chloride in their sweat, and also some present pancreatic insufficiency. So to this date, more than 2,000 mutations have been described in the CFTR gene. And these mutations have been classified into teratypes, which are classes of mutations that can be rescued by the same drug, so by the same therapy. Here in the field, we call them CFTR modulators. So, and just to give you some examples, read-through compounds were suggested to rescue some mutations that give rise to premature stop codons. Correctors were actually approved to rescue some class 2 mutations in which there's no transport of CFTR to the apical membrane. And for example, potentiators have been approved for class 3 mutations, which are mutations that lead to impaired gating, meaning that the protein reaches the apical membrane, but it does not function well as a chloride and bicarbonate channel. So the current therapeutics that patients with cystic fibrosis have are actually this triple combination of 2 correctors and 1 potentiator that has been approved for homozygous and heterozygous of the F508del mutation, which is the most prevalent mutation in these patients. Also, Kaleido, which is a potentiator, has been approved for some gating mutations and some residual function mutations. But there's this group of patients that bear very rare mutations that are often excluded from clinical trials due to the low representativity of their of each individual mutation. So they lack any approved modulator, so they don't have any therapeutic option regarding CFTR modulators. And this is where the HEAT-CF project comes along. So this project has two main stages. So the first step was the collection of 500 biopsies in European individuals with cystic fibrosis. So then after collecting these rectal biopsies, we isolate and cultured intestinal organoids, which were our in vitro model, to test drugs from 3 pharmaceutical companies and to predict the clinical benefit of each one of these patients. So the second part was the selection of the best responders per drug. And then the end of this project will be the a clinical trial for each one of the pharmaceutical companies. So using the best responders. So the model that we are using in this project and that we want to show that it can be used as a new biomarker in cystic fibrosis are the intestinal 3D organoids. So these are cells that can be isolated from rectal biopsies, in which we can isolate the intestinal crypts that have a niche of stem cells that can be cultured and expanded in a cell culture. And we can establish these intestinal 3D organoids that are these cells here, using some appropriate medium and a 3D matrigel dome. And then these cells which express or that highly express CFTR can be used in a drug response assay, which is called the forskolin-induced swelling. So as the name says, we use forskolin, which is a compound that activates CFTR to induce this organoid swelling. So it means that when we use or when CFTR is rescued by a combination or drugs, there's a transport of fluid to the inside of the organoid, and so their area increases. So for example, here we have an example of F508del homozygous patient that, using a combination of a corrector and a potentiator, their organoids increased in size. So it means that the mutant CFTR was rescued. So this project started by the recruitment of 502 patients throughout Europe, and we also were included. So we have 13 patients from Portugal that were included in this project. And this is a collaborative work from 4 different labs that, so 3 are academic labs that have never stopped working even during the confinement in March. So it's our group from BioISI, a group from the University of Leuven in Belgium, and the University of Utrecht in the Netherlands. And we also have an industry partner that, along with the 3 of us, so we made the organoid team, let's say. So since the aim of this project is to find alternative treatments for these patients with rare mutations, we combine the efforts with these 3 pharmaceutical companies that have some CFTR modulators in their development pipeline. And in this way, they can actually predict their own products' efficacy, so their pre-drugs efficacy using the samples from these patients. So we are working with 3 different companies. I'm here only showing 2. So we work with proteostasis therapeutics that have currently a potentiator, a corrector, and an amplifier in their development pipeline. An amplifier increases the amount of messenger RNA. A corrector transports the protein to the apical membrane of the cell, and a potentiator increases their opening probability, so their function. So Elox Pharmaceutical is the other company, which is actually more focused in stop mutations. So their read-through agent, so it's a compound that actually reads through the premature stop codon and allows for the production of a full-length protein. So I'm just going to show you some preliminary results in our primary screen. So we already screened all of these patients using a compound from one of the pharmaceutical companies. And we showed here that there are some non-responders as expected. We also saw that there was a group of atypical CF patients, so meaning that here in the DMSO conditions, they already have a high residual function, so even without the drugs. But what is more interesting and exciting is this group of responders, so individuals that have increase of CFTR rescue and activity upon the incubation with these pre-drugs from the pharmaceutical companies. So just for you to have an idea of what the assay looks like, these are the organoids in the control situation, so with no drugs other than forskolin, and they don't increase their size. But upon the incubation with the drugs that we are testing from these pharmaceutical companies, you can see that there is this increase in the organoid swelling. So this means that we have high responders to these new drugs. So this project mainly has led us to some, or opened the doors for some opportunities. So in the end, we want to show that we have a new model to predict clinical benefit for these patients. We also will show that the intestinal organoids can be used as a tool for preclinical patient stratification and drug repurposing. We will also, or one of our aims will also to get the off-label extension of drugs that are in the pharmaceutical pipelines. We will provide the clinical trials for these patients that have rare mutations that otherwise would never have this opportunity to be in a clinical trial. We will also start this biobank of intestinal organoids, so cells that can be used for research and for future drug development. So these intestinal organoids can be biobanked for many years and be always available to put again in culture when new drugs come along. And so in the end, we just want to increase the knowledge on personalized medicine in the field of cystic fibrosis by using each individual cells and to predict each response to every drug that is available. So I would like to thank to my group here in BioISI, to our collaborators, also to the pharmaceutical companies and to the European Union for funding, and all of you for your attention. Thank you.
Thank you very much, Iris. And I think it was particularly nice to end with a project that so well translates what I said in the beginning of how fundamental research can now translate so quickly into an application, in this case, the personalized medicine, and the fact that patients can have access, easy access at a much lower cost.
Yeah. Exactly.
Any immediate question to Iris? Or, as I'm going to say right now, in order not to delay the session that will follow significantly, which was supposed to start now, I propose that we make, rather than 15 minutes, only a 5 minutes break, and then we return for the next session, which will be shared by my colleague Paul Costa. and some of the selected highlights in terms of outputs of BioISI in this year, so 2020. Once more, thanks a lot to all the speakers, nice presentations, and to everyone that has been attending this BioISI Research Day. Thanks a lot, and I'll see you again in 5 minutes. Okay, thank you.
Hello? Hello? Good afternoon. I don't know if everybody can hear me.
Yes, definitely.
Okay, I think it's time to start the new session, and this session is about we are going to present selected BioISI papers, and these papers were selected according to the 5 thematic lines which we have in BioISI, and it intends to highlight the research done on those thematic lines. Although I think most of these presentations will show that there is a large intersection between those thematic lines. So without further ado, I will give the screen to Margarida Quaresma, which is going to present mutant CFTR drives twist-1 mediated epithelial mesenchymal transition. I'm sorry. Go ahead, Margarida.
So can you hear me okay?
Yes.
Okay, so thank you for this opportunity to share my work. So it's good that I'm actually after my colleague Iris, because now you heard about cystic fibrosis. So today I'm going to talk to you about our recently published paper, mutant CFTR drives twist-1 mediated epithelial mesenchymal transition. And I will just give you the highlights, but if this interests you, I highly recommend you go check out the complete work. So as my colleagues have already mentioned, cystic fibrosis is the most common life-shortening monogenic condition in Caucasians. It's a multi-organ disease, so the patients have a lot of symptoms, but we focus mainly on the airways because pulmonary insufficiency accounts for 80% of CF mortality. And this disease is caused by mutations in CFTR, which the gene encodes an ion channel expressed at the apical surface of the epithelial cells in a wide variety of tissues. And these mutations in CFTR can produce different defects, as Iris already talked about a few of them, but the most prevalent mutation is this F508del, accounting for 80% of patients. And it actually impacts CFTR traffic and its stability at the plasma membrane, and this is the one that I'll be focusing more today. So the other concept that I need to introduce to you is epithelial mesenchymal transition, or EMT for short, which is a developmental process which involves transcriptional reprogramming of epithelial cells into mesenchymal cells. So essentially, the epithelial cells, they undergo several processes that change them completely phenotypically. So basically, they lose their junctions and their adherence to the matrix. They lose their cytoskeletal architecture, they lose their polarity, and eventually they gain invasive or migratory properties, and they can proliferate more. This process is very important in development, as I said, but it can also be reactivated in pathological conditions like inflammatory diseases and cancer. And what I need you to take from this is basically the simplified version, right? So during an EMT, what happens is a decrease in the epithelial markers with an increase in mesenchymal markers and also an increase in the so-called EMT transcription factors, which are the main players that regulate this process. So the cells go through a variety of stages, and they can be fully epithelial or having a partial phenotype or mesenchymal. Now, the thing that you also need to know is that CF is also an epithelial differentiation disorder. So what happens in CF cells is that they have impaired or delayed epithelial differentiation, increased cell proliferation, loss of cytoskeleton organization, and effects in the tight junctions and barrier function. So actually, this model should look a little bit more like this. So this channel is actually impacting a lot of different aspects of the cell. And actually, a transcriptome profiling meta-analysis performed by our group revealed an EMT signature in the airways of individuals with CF. So is it possible that EMT is an active process in the CF airways? Basically, there are a few clues in the literature. So CF individuals display developmental defects. So the lungs and the male reproductive tract have compromised structure and function. Downregulation of CFTR has also been shown to lead to EMT and fibrosis. And CFTR has actually been identified as a tumor suppressor protein and an EMT suppressor in a cancer context. So this led us to propose this model in another work by our group, that actually CFTR has roles in development, in recovering after injury, and preventing fibrosis, and actually in preventing against cancer stimulus. But the question remains, is EMT an active process in the CF airways? And so this was what we tackled in this paper. So basically, we began by looking at human native bronchial tissue. It's as close as it gets to the, the patient situation. So these are CF patient lungs and control lungs. And we probed for epithelial and mesenchymal markers. And right away, what we could see was that the epithelium looks different. So a healthy epithelium has this columnar cylindrical shaped elongated cells, is a pseudostratified epithelia. But this appears to be lost in CF. Actually, we can see several cell layers here, which is not supposed to happen, and it's indicative of disease. We could also see disorganized or disrupted proteins. So ZO1 is a tight junction protein. It's important for the barrier function of the epithelia here in the apical side, but we can see here in CF lung that's very disrupted and dysregulated. And we could also see that the mesenchymal proteins were increased in the patient lungs. So basically what we found was that the CF epithelium was disorganized and showed upregulated mesenchymal markers. So this is partially in agreement with an EMT signature. So then we went to check primary human bronchial epithelial cells, which are also a very physiological model. And we checked both epithelial and mesenchymal protein levels. And what we could see, and this applied to cells with different genotypes, so the mutations have different effects on CFTR, but we could see in all of them an increase in the mesenchymal protein levels. And this was the case even when some of the CFTR was reaching the plasma membrane. So as it's represented by this band here, which is the mature form. So even when this happens, the mesenchymal markers are still increased. So to try to get a bigger picture, we wanted to see what was happening with the so-called core EMT transcription factors, which define the EMT signature. So we went back to lung and we did a transcriptomic analysis, and we could see that almost all of these major EMT transcription factors are increased, and TWIST1 and ZEB1 particularly are significantly increased in CF patients. And we could also confirm this in our primary cells. Both TWIST1 and SNAIL1 and SNAIL and SLUG were increased. And also in our immortalized cell models, we could see this particularly for TWIST1, which is significantly increased when we express the mutant CFTR form. So essentially, we can see that mutant CFTR leads to an increase in mesenchymal proteins and EMT transcription factors without the loss of epithelial protein expression. So these are hallmarks of an active partial EMT. So to further confirm this, we took our cells, we polarized them to make them more physiological, and we treat them with correctors that help to recover the traffic to the plasma membrane and potentiators that increase the function. And so this is what we did. So we treated our cells and we checked for epithelial and mesenchymal proteins. And basically, what we can see here is that the correctors are exerting their proper function. So they are rescuing F5-Voigt-Del CFTR to the plasma membrane. And this effect was sufficient to significantly decrease the mesenchymal protein levels. So there is a correlation between the correction of mutant CFTR and the significant reduction in these mesenchymal marker levels. So actually the amount of functional CFTR is closely correlated with the epithelial or mesenchymal status of the cells. So to go a bit further, we treated our cells with an EMT inducer, TGF-beta1, and we checked what was happening with the epithelial and mesenchymal markers. So what we can see is that in the wild-type CFTR cells, there is a very small drop in ecadherin level. This is an epithelial protein, whereas in F5-Voigt-Del, there is actually 63% drop, so much more steep. And regarding the mesenchymal marker vimentin, we see no effect on wild-type CFTR cells, but when we treat with TGF-beta1, there's a 10-fold increase. So we can already see that wild-type CFTR expressing cells are more resistant to the induction of EMT than CF cells. And then we wondered if we could make this a little bit better by using, again, our correctors and potentiator. And so this is what we did. You can see here the F5-Voigt-Del corrected here. And what we can actually see here is that by potentiating wild type, there is a complete block of the TGF-beta1 mediated DMT induction, so complete block. And here in F5-Voigt-Del, the rescue can actually partially block TGF-beta1 mediated DMT induction. So this again suggests that CFTR is quite important in protecting against this EMT. So then we wanted to see if we could pinpoint the mechanism by which this is happening. So if you remember, TWIST1 and ZEB1 were significantly increased in lung, and ZEB1 was not expressed in detectable levels in our cell models. So we wanted to check TWIST1 and SNAIL1 to see if we could pinpoint the mechanism that was, affecting all of this. So we went again to our cell lines treated with the correctors and the potentiators. And what we could see right away was that correcting F5-Voigt-Del CFTR showed a specific and significant reduction in TWIST1 levels while having no effects on snail and slug levels. So this again seems to point that TWIST1 might be the specifically link between CFTR and EMT induction. So then what we did was we treated our cells with an SH to silence TWIST1. And what we could actually see was that the TWIST1 knockdown was sufficient to block the vimentin expression that we were seeing in previous experiments. So this knockdown was, sufficient to block partially EMT in these cells, suggesting that TWIST1 might be actually the player involved here. So in this work, we could see that a partial EMT is active in CF airways, that mutant CFTR leads to an increase in mesenchymal markers and EMT transcription factors, but without the loss of epithelial protein expression. However, the CF epithelial cells show disorganized epithelial proteins. And we could see that this partial EMT phenotype could be directly related to the expression of mutant CFTR. But we could also see that rescuing this mutant CFTR to the plasma membrane could revert this mesenchymal phenotype. And we concluded that CFTR expression and function are very important in protecting epithelial cells against EMT. And lastly, we concluded that mutant CFTR is driving EMT through a TWIST1 pathway. So essentially, we did confirm that CFTR plays a major role in maintaining epithelial cell differentiation, and its dysfunction is enough to generate partial EMT. So this is just a final model summarizing our findings. So this is a healthy epithelial cell with CFTR here in the plasma membrane. It's protected from detrimental stimuli, but in CF patients, they don't have the CFTR protein here, or they have less. And the cells are already in a more mesenchymal state right away. And then actually as the CF lung disease progresses, the CF lungs, they have an environment that primes the cell for further aberrant EMT progression. So they are already sensitized. And so this is probably why, we see patients with such higher cancer incidences and other defects. So this is it from me. I would like to thank my group and my supervisors and also the colleagues that participated and helped me write this work. And yeah, that's it.
Thank you very much, Margarida. I'm sorry, but I forgot to mention that this was the paper selected from the biomedicine thematic line. This is important. And there are any questions? Anyone?
Yeah, I have one.
Okay, go ahead.
Sorry. Hi, Margarida, how are you?
Hi, I'm
very
good.
Nice work and very nice presentation. Just a quick question: when you did the TGF-beta reverse TGF-beta effect reverse experiments using the correctors,
yes,
did you use all correctors in the wild type cells or just the potentiator?
All of them.
All of them. Okay.
Yes.
But did you try to use the drugs independently?
Not in that particular experiment. In other ones, yes, we did. You can check, I think it's figure 5 of the paper, where we used only the correctors and only VX661 separate. And you can see actually that VX661 is not enough to really put a lot of F508del CFTR in the plasma membrane.
But I was asking you about the wild type protein, because you show also that the drugs had an effect in the wild type cells.
Yes, we did everything in parallel in both cell lines always.
Okay. I'll check it later. So
yes,
thank you.
Okay, thank you very much. Let's move along, and now it's time for Manuela Costa. This is the highlight from the biotechnology thematic line, and we are going to see Manuela's presentation. I think it's a recorded one because Manuela could not be present. So if you have any questions, you can post them on the chat, and then Manuela will try to answer them. So I think Simo has to put the video.
Hi everyone, I'm sorry I can't be with you today, but I would like to share the work we are doing in the University of Minho in Braga. And we have been working with flower development of Quercus suber, which is this amazing tree from which we extract the cork from. Not only is very important for the economy of our country, but there's also, there are also very interesting biological problems to be studied. And this year we have published 2 publications, one on genome-wide identifications of epigenetic regulators, and another one on the molecular mechanism that controls flower development. And some of these results I'd like to share with you. So this species is monoecious, which means that has male and female flowers in separate parts. And the male flowers emerge around March and in the axils of the buds when the buds break. And to see the female flowers, you need to wait another 4 to 6 weeks. So there's a temporal and spatial separation of the female and male flowers. There is also another very interesting thing in this species, which is the pollen gets deposited in the stigma and it germinates a bit, and then it stops for 1 to 2 months, and only later it gets fertilization occurring. And then you see the fruit emerging in autumn. But some Quercus species, and indeed some of the trees of Quercus suber, have biennial fructification. This means that fertilization only occurs 1 year later, so that the pollen stays stationary for 1 year. And what you see, for instance, is that you see some in the autumn, you see some fruits that are the results of the fertilization of flowers that were formed 1 year before, and you see other flowers that will stay dormant during the winter. And because this is an arrested state during the winter, it is possible that it may be controlled by the deposition of epigenetic marks. So we have been studying the deposition of marks associated with active and repressive states of chromatin during differential stages of flower development after pollinization up to fertilization in annual and biennial flowers. This is ongoing work in the lab of our collaborator of Leonor Moraes, and soon we'll have more to tell you about this. But we also have identified epigenetic modifiers, DNA and histones, in the genome of Quercus super, and we use transcriptomic databases to understand their expression in different tissues and conditions. But to understand better what's going on in biennial fructification, we are actually studying a species that's strictly biennial. This is Quercus ceres, and we obtain a transcriptomic profiling of female flowers after pollinization, with actively growing, stationary, dormant during the winter, and we are currently exploring these differentially expressed genes. Actually, you got interested in working with these species much earlier on when you got together in a consortium nationwide. That the idea was to get a transcriptome for these species, and at the time we unveiled many differentially expressed genes in female and male flowers at different developmental stages, and among these genes there were genes of the ABC model, which is very quickly a model that postulates that flower organs are the combination, are specified by the combination of different proteins. And for instance, stamens, you need class B proteins together with class C and E, and to have carpels, you need class E and C. So we went and looked along our transcriptome for these genes being expressed, and actually E and C class genes, there is no difference between male and female flowers in expression, but the B class genes are much highly expressed in male flowers, and actually one of them, Pistillata, is a very good marker for male flower development because there is no expression in female flowers. So we can actually check the functions of these genes because we can complement mutations in Arabidopsis with the Quercus genes. And for instance, Pistillata is a mutant that has no petals or stamens, and when introduced to Quercus genes, the phenotype is complemented, meaning that there is functional conservation of these genes. So we did this for all of them, but just showing here the example for Pistillata. We also, in a combination, using a his2 hybrid, we checked the ability of these genes to dimerize, and at the time we proposed a model that can explain why different protein interactions between these genes can specify male and female flowers, and you can check our publication for more details. So we were also very interested not only in understanding how these flowers develop, but how they are induced. Remember, they are induced, they emerge at slightly different times during the year. And we'd like to pinpoint the time in which flower development is initiated. So during the winter, you have the buds that stay dormant, and then in the axils of these buds at bud break, you have the emergence of male flowers. Female only emerge a few months later or a few weeks later in the axils of newly formed leaves. Okay, so you need to have new leaves being formed in order to see female flowers emerging. And then you have again the formation of new buds that will stay dormant during the year. So what we did, we went and checked on genes that code for repressors known to be involved in dormancy in other species or in induction of flower, and we checked the expression in the buds between bud set to bud break. And what you saw was that at bud set, you have high expression of genes that are normally associated with dormancy, which is expected, and then expression goes down. And at bud set, you have high expression of the genes that are known to be activators. Actually, you can check for function of these genes by introducing them into the Arabidopsis. And some of these genes, for instance, FT and SPL4, induce flower early in Arabidopsis, whereas other ones repress flower. We were actually quite puzzled by one of these genes that are an activator that was highly expressed in the bud in September, as if it was associated in bud set, whereas the other gene that is known to be the prime activator inducer of flower, which is the FT, is only upregulated in the bud in March. So FT actually is the florigene, is normally expressed in the leaves. and therefore we checked to understand better this expression of this gene in leaves throughout the year. And we were extremely surprised because there's 2 peaks of expression of FT, and this has never been seen in any other species. Usually there's a peak of expression of FT and flower gets induced. So could this be that this species has 2 peaks of flower induction? And to check this, if this is the case, we wouldn't be able to see these 2 peaks in plants that didn't flower. In juvenile trees, for instance, we don't see the expression of these genes at all. So we were quite convinced that this is more related to flowering. And one thing could be that maybe there is flowering occurring in March, induction, and then in August. And could it be that inside this bud you have the formation of flowers already, the ones that are induced in August? So we checked for expression of the genes that are expressed typically in flowers. And in particular, remember, pistillata was a very good marker for male flower development, and we could see the expression by in situ hybridization in the buds of September. This means that in September you have already flowers inside, male flowers, whereas in the buds of March you don't see the expression of pistillata, but you see expression of other genes that are specific expressed in flowers. So our model is that actually you have the expression of FT in the leaves in August, and then you will have the expression of genes that will induce flower development in the buds. Male flowers will stay dormant during the winter, and then they emerge at in early spring. And then you need to have a new second peak of expression of FT in order to induce the female flowers inside the bud. So we think there are 2 different stages of flower induction in this species, which is quite a new thing to be reported. So I hope to have given you a good overview between what induces flower, what makes the flowers different, and what makes events that may justify the difference of annual and biennial fructification. And thank you for your attention. And I would like to thank people for actually doing this work, my collaborators and the people in my lab. If you are interested in knowing more, drop us a line, visit us in Braga whenever possible, and we'll be delighted to talk to you. Thank you.
Okay. As I told, Manuela is not here, so you can leave your questions if you want. And now we can proceed to Mário Rodrigues, which is going to present the highlights from biophysics thematic line. The title is Mechanical Properties of Human Bronchial Epithelial Cells Expressing Wild Type and Mutant CFTR. So please, Mário.
Olá, boa tarde. Conseguem ouvir-me?
Sim.
Sim, ok. Portanto, partilhar aqui a apresentação. Share. Portanto, mais uma vez, boa tarde a todos. Portanto, eu vou apresentar o trabalho Mechanical Properties of Human Bronchial Epithelial Cells Expressing Wild Type and Mutant CFTR. So I'm Mario. I'm going to be presenting this work, but actually the expert on cystic fibrosis is Carlos Farinha from the Functional Genomics and Proteostasis group. So of course, all the people are involved. I guess you need to feed the cells, and you need to measure them, and so on. So first, just a few notes. Well, you'll see that my presentation will have much less genes and proteins than the previous presentations. Okay, so first I would start to say why mechanical properties. Of course, I guess it's pretty obvious that mechanical properties are involved in a plethora of key mechanisms from migration to drug cell interactions, and additional notes that I would like to make is that so currently there is a huge interest in measuring mechanical properties because they are fundamental to explain the cell structure, evolution, and response to different stimuli. However, the accurate measurement of mechanical properties remain a difficult experimental task for different reasons that unfortunately I will not have time to address. Another important issue is that a cell is a very heterogeneous body, and so its overall complexity makes it really hard to correlate mechanical properties with the microstructural details of the cell, even though these correlations are needed if we want to move towards better understanding of mechanobiology. Now, in our laboratory, we've been very much involved in the accurate measurements of these mechanical properties by developing different protocols and trying to tune these protocols to make better measurements. And in the future, we intend to move forward towards a prediction of mechanical responses from known cell features. For example, you make a fluorescence microscopy images of the cytoskeleton, and then from differences in the cytoskeleton, are we able to predict mechanical properties? Okay, so I mean, I've seen already a few talks on cystic fibrosis, so you know that most of the times this disease is caused by the deletion of the phenylalanine amino acid, and of course, disorganization of the cytoskeleton is important to place the protein at the right position to do correctly its job. So there has been previously reported. It has been previously reported that this disorganization has impact on the disease. So in this work, we'll investigate the mechanical properties of bronchial epithelial cells transduced with either wild type or the deletion of the amino acid. We will be using both conventional AFM, because it's conventional, and the technique developed by us, which we call force feedback microscopy, and it has been the focus of Miguel Vitorino PhD thesis. He has been tuning this experimental apparatus and making it work well on the measurement of mechanical properties of cells. So just to make sure that we all speak the same language, which is sometimes not as easy as it may seem, given the interdisciplinarity of our institution. So we are going to be concerned with what we call the Young modulus and the Poisson ratio. So very briefly, if you stretch something, imagine, for example, a rubber band. If you stretch the rubber band, you must apply a certain force, and then this force will be related to the strain, or so to the deformation by the Young modulus. And you've probably seen that when you stretch a rubber band, the rubber becomes thinner, and that is because it tries to maintain its volume constant while you are stretching. And that is given by a parameter which we call the Poisson ratio. So for example, if I go to the previous talk where we were discussing cork, if you put a cork on a bottle, the diameter shrinks, but the length of the cork remains the same, and this corresponds actually to a material with the 0 Poisson ratio. So, of course, a cell is not such a simple object as the one I depict here. So in practice, we are never going to measure neither the Young modulus nor the Poisson ratio. We are always going to measure a combination of these two parameters. I must say that so these two parameters completely characterize a material when the forces applied are small, so the deformations are small, and when these deformations are performed very slowly, so that there is no dissipation mechanisms, production of heat or whatsoever. Also, the Young modulus is quite a complex thing. Even in simple materials, it depends on direction. So in a cell, you can imagine that is really much more complicated. So how do we usually infer we are going to actually measure a combination of Young modulus and Poisson ratio, and so we're going to measure what we call the reduced Young modulus, and the reduced Young modulus is defined here on this expression 2, and it involves the mechanical parameters of a body with which we ident a cell, and it also involves the mechanical parameters of the cell itself. So then the way you do this is that you usually approach a cantilever that can deflect. You may put a sphere at the apex of of a very sharp tip, or just leave the very sharp tip as is, and then you press against the cell. The cell is going to deform. The deformation is given by what I call indentation and is represented by this delta. Well, I will skip this equation below because otherwise I will not have time to finish in time. So in certain simple conditions, the force that you apply as a function of the indentation is given by this very simple expression. So this is what we call Hertz's model. It assumes that there is no adhesion. It might seem odd, but actually these experiments are done in buffer, and the buffer shields the attractive forces, and so the adhesions are really very small, and when that is the case, we have this very simple expression. Now, of course, to accurately measure the Young models, which is what we want to know, you need to measure the force and the indentation. This is not measured directly because when you squeeze the cell, there is going to be two deformations. So one deformation of the cell and deformation of the cantilever, and these two are combined. So you have to find the means to disentangle these two deformations in order to use this very simple expression. Now, just to give a very short summary on the differences between conventional force microscopy and force feedback microscopy. So, well, on black, I highlight the advantages. So force microscopy, one of the advantages is conventional. I see that as an advantage because when you are going to publish things, you may bias referees in different ways. If you provide them with a technique that they have never seen, they might think that you are just crazy. Then there are other advantages of conventional microscopy. You buy it and it's very easy to use. Force feedback microscopy, you cannot buy it, so you need to develop. You need someone like Miguel Vitorino for that. On the advantages side, well, force feedback microscopy, so the calibration is very reliable because it's done with an interferometer. So this calibration is well, virtually error-free. Whereas this calibration, well, I will not have time to go into the details, but it involves a laser and a photodetector, and it's quite cumbersome. And well, you can go through the literature and you see there are many articles talking only about the fact that the calibration is unreliable. Then another very important difference is that in conventional force microscopy, the tip is free to deflect on the cantilever. And so you have the combination that I said before of a sample which is deforming and the cantilever that is deforming. Whereas in force feedback microscopy, you use a mechanism to avoid the tip from moving. So when you actually do a measurement, you directly measure the indentation. So you directly probe the very simple expression that I showed before. So then when to further highlight this, so in conventional AFM, you measure a photodetector voltage, so which is a voltage that depends on where the laser is incident on a detector. And then you, of course, you have to calibrate these to go to a force. And this calibration, as I said, is very error. It's very likely that you make an error, but if it is, say, a 20% error, you're going to get a 20% error here, say, let's assume that is not very bad. However, you measure vertical displacement, but what you need is the indentation. And in order to obtain the indentation, you need to subtract from the vertical displacement, you need to subtract the deflection of the cantilever, which is embedded here. And this, as I said, is not error-free. And then when you convert, you have to mix these two axes in order to obtain this third axis. And when you do that, well, the error propagates, and you can actually get imaginary indentations, well, things that don't make sense at all. So another way to illustrate that is that in force feedback microscopy, you directly measure these, whereas if you would try to make an expression for the voltage Z that you measure on the photodetector, well, it wouldn't fit the next two slides, really. I'll skip that one. So going now to the results. So what we do, well, we have on the left one example curve of data obtained with conventional microscope, and on the right with the force feedback microscope. And of course, this data is already, well, displacement has already been converted to indentation. So all the troubles have been solved. And well, this is to obtain the young modulus. What you do is that you take several tens of these curves over a cell, and then you compute an average for each cell, and then you repeat that on many cells, and eventually you will get an histogram like this. So on the left here, I show the results we obtained with the force feedback microscopy. Notice the very small error bars that we have. And on the right, the results obtained with conventional atomic force microscopy. Now, one conclusion already is that the wild type cells present larger young modulus, and this is true in both cases. One difference you see is that the young modulus as determined with the force feedback microscopy is actually smaller than the one determined with conventional microscopy. This might be, well, eventually due to systematic errors that occur when you use conventional microscopy, but given the large number of cells, and we have excluded also many cells that have a very odd young modulus. So we guess the reason is not that. So we believe that this is the fingerprint of the cell's heterogeneity. So these very sharp tips that we have used with the conventional microscope, they have, well, apex radius of about 1 nanometer. So you actually probe the heterogeneity of the cell, whereas these spheres have about 1 micrometer. So you can imagine, since the cell is not homogeneous, you can imagine that the cell has points which are harder, and so the actual contact surface when contact with the cell is actually much smaller than this 1 micrometer sphere. So basically we are overestimating the contact surface, and that results in a smaller young modulus. This is something we would like to check in the future, but we guess it is just a signature of the cell's heterogeneity. So in conclusion, we used both atomic force microscopy and force feedback microscopy with a sharp or a spherical tip. In both cases, we found that the expression of mutant CFTR causes a decrease in the cell's apparent elastic modulus. And finally, we hypothesized that this decrease is likely the consequence of a more disorganized actin cytoskeleton, since the cytoskeleton is the part of the cell which more, well, the young modulus depends mostly on the cytoskeleton properties. And well, as perspectives, so we would like to quantitatively understand why the elastic modulus seem lower with the probing with the sphere. So we have planned a few experiments to understand that, and then we will move on forward to probe cells and test for different correctors and potentiators that will help to place the protein at the right position where it should be to do the function it should do, and then see whether that has an effect on the mechanical properties of the cell. And lastly, we will, if we find that there are mechanical differences, we aim to quantify rather than just measure these differences. And well, then I would like to thank BioISI, of course, for putting these very different people and interests together. And thank you too, and sorry for the interruption before. So if you have any questions.
Thank you, Mario. Any questions? You can also post them on the Zoom chat. I don't know if there are any questions.
One very short question, Paulo.
Yeah. Go ahead, Rui.
Would not be to Mario, but anyone, to Mario or Carlos, because I'm sure there's with this mutation, there's probably already data on the actin cytoskeleton to confirm or dismiss this hypothesis. Is that
Sorry,
so?
can you repeat the question? If this
there's probably already some cell biology data of actin cytoskeleton in these cells, in this mutation, that could tell and confirm if there is a disorganized cytoskeleton or not. Is there?
I've just joined, Mario. Do you want me to answer?
Yeah, sure.
Okay, so I missed I missed the beginning of the presentation because I just came out of a PhD thesis from ITQB, but I entered around slides 5 or 6. So yeah, there are reports that the cytoskeleton organization and the link of PDZ proteins that stabilize CFTR at the plasma membrane is different between the wild type and the F508del CFTR.
Good.
And yeah.
Okay, so let's proceed now, I think, in the sake of time. And now we are going to hear the talk of Hugo Martiniano, which is the selection from the bioinformatics thematic line. And the title of the talk is Identification of Biological Mechanisms Underlying a Multidimensional ASD Phenotype Using Machine Learning. Hugo, go ahead.
First of all, I would like to thank the opportunity to present this work titled Identification of Biological Mechanisms Underlying a Multidimensional ASD Phenotype Using Machine Learning. I would like to begin by addressing the motivation for this work. The complex genetic architecture of autism spectrum disorder and its heterogeneous phenotype make molecular diagnosis and patient prognosis very challenging tasks. To establish more precise genotype-phenotype correlations in ASD, we developed a novel machine learning integrative approach, which seeks to delineate associations between patients' clinical profiles and disrupted biological processes, inferred from their copy number variants spanning brain genes. This scheme outlines our integrative systems medicine approach to identify complex genotype-phenotype associations. This study, we used clinical and genetic data from the Autism Genome Project. Clinical data comprises reports of ASD diagnosis and neurodevelopmental assessment instruments. Genetic data consists of copy number variants, from which we selected for analysis rare, high-confidence CNVs targeting brain-expressed genes. There are 4 main steps: clinical data processing, genetic data processing, and 2 machine learning steps that combine clinical and phenotypic data to establish genotype-phenotype associations and predict clinical outcomes of ASD patients. Agglomerative hierarchical clustering was used to identify clinically similar subgroups of individuals in stable, validated clusters defined by multiple clinical measures. Clustering analysis identified 2 distinct phenotypic subgroups, Cluster 1 and Cluster 2. Patients in these clusters differed significantly in ADAS-defined severity, adaptive behavior profiles, intellectual ability, and verbal status, the latter contributing the most for cluster stability and cohesion. These 2 clusters are broadly characterized by a milder and a more severe phenotypic presentation. Functional enrichment analysis of brain genes disrupted by rare CNVs identified 16 statistically significant biological processes, including cell adhesion, neural development, and cognition, in line with previous ASD findings. The values of mean decrease in accuracy obtained from random forest categorization of subjects into ASD phenotypic clusters offer a complementary view on the role of disrupted biological processes in ASD. We showed that the most statistically enriched biological processes are not necessarily the ones which allow better patient classification. A naive-Bayes classifier trained to predict the ASD phenotypic clusters from the 15 disrupted biological processes achieved low prediction ability for all clustered individuals. However, for a subset of patients with higher biological information content scores, predictions with a high precision but low recall were obtained. This indicates that milder and more severe clinical presentations can have distinct underlying biological mechanisms and that genotype-phenotype correlations can be established, at least for some individuals with ASD. In conclusion, we've developed a novel integrative system biology approach to predict the severity of ASD from biological processes targeted by genetic variants. ASD patients were clustered in two subgroups, characterized by milder and more severe phenotypes. This is consistent with results from the literature. Overall, the present approach is proof of concept that genotype-phenotype correlations can be established in ASD, and that biological processes can predict multidimensional clinical phenotypes. However, predictions are strongly dependent on patients' information content. These findings are therefore a first step towards the translation of genetic information into clinical useful applications. Thank you for your attention. I'm at your availability for any questions.
Okay, thank you. I think Hugo is here to answer some questions if there are any questions. Okay, I might have one. You mentioned in the very end that this is just the first steps towards probably implementing this in probably routine assessment of ASD. How far do you think it is? I mean, how far do you think it will have a clinical practical application?
Hello, Paul, and thank you for the question. I mean, I'm not. I wouldn't say this is very near to really an application in the clinics, because I mean we need a lot of data in order to train these models and to apply them. So this is this data set is comprised of over 2,000 individuals. So
you would say that the limitation is you would say that the limitation for now is the collection of data. You need lots of data to really train a good model. Is that it?
That and the ease of collecting patient data, because going from assessing the patient to collecting all the data necessary to apply this is not easy. So that's one of the main limitations. It's mainly a technical on the clinical side limitation.
Okay, thank you. And let's see if we have any further questions.
Paulo, can I make a
Nuno,
question?
Go ahead.
Hi, Hugo. Thanks for the really nice presentation. I just wanted to ask you whether you guys tried, I guess you didn't, but whether you know if this is an approach where you actually train your models using data from people without autism and people with autism, and from there you try to find some kind of pattern or something, because if I understood correctly, your data set is only from patients. Is that correct?
Mm-hmm. Yes, exactly. So in this case, we don't use any control data. So actually the first step is to try to define what is a milder and more severe autistic phenotype, and then the classifier just tries to discern both subgroups. So that's also another issue, is that it's not very easy, at least the clinical part, to get patient data, to get the controls.
So if you entered if you had that data available, which I guess it's not that simple to have good quality data, do you think that the kind of model you guys are using could distinguish fairly well between people that are completely healthy, let's put it this way, I don't know how this terminology is normally used, and people who have some degree of autism, and you can even go to the extreme cases, I mean very severe forms of autism versus people that have absolutely no autism?
Yes, I mean, for very severe cases, it definitely should be able to create a model to discern in both cases. For the less severe, it's not. I mean, not so easy to answer.
Yeah,
we would have to try.
All right, thanks a lot.
Mm-hmm. Welcome.
Okay, so let's move on now, and now it's time for Elizabeth Silva, which is going to present the highlights from the biological chemistry thematic line, and she's going to present a work entitled Biofouling Inhibition with Graft Econia Biocide towards a non-releasing eco-friendly multi-resistant anti-fouling coating. Hi, Elizabeth.
Hi, good afternoon. Thank you, Paulo. Good afternoon to all. So today I will speak a little bit about biofouling burden in industrial systems. And first of all, what is this biofouling burden? And here exemplified for aquatic systems. Well, this biofouling is no more than the natural and spontaneous colonization of micro and microorganisms on submerged surfaces or surfaces in contact with water. For example, cooling water industrial systems or water treatment stations, or even for the marine industry to transport ships. So this biofouling, as you can see here in this image, if no measure is taken, in just a few days, you can reach this macrofouling level. So this macrofouling can lead to serious problems, for instance, clogging of the system of pipelines or filters, or even promote biocorrosion, premature corrosion of the materials, or in case of shipping, can increase significantly the drag fishing increases. So huge maintenance costs arise from this biofouling, as well as, for instance, in the next slide that I will show you, in the case of the shipping industry, only for transport delays, not account with maintenance costs, could lead to billions of euros per year spent. Moreover, this kind of problem can also raise the number of waterborne outbreaks. As you know, in Portugal, you heard the problem of Legionella disease. So Legionella came out with the problem of this biofouling. So unsuccessful cleaning or a successful prevention of this biofouling raise these outbreaks. So of course it is a universal problem and known, and so there are viable anti-fouling solutions. But as I can see in this slide, these viable solutions also pose a new problem, an environmental problem. Why? Because they act by the releasing of toxic chemicals, or directly by disinfection or cleaning, or indirectly through anti-fouling coatings. So, and still, as an example of marine industry, 90% still use these toxic anti-fouling agents. For instance, copper-based ones, and millions of liters are still released to the waters. So these came out with top priorities world and are included in specifically in sustainable development goals. And the main goals is to at least save our waters from chemicals, and also to provide sustainable systems for these industrial sectors. Otherwise, we couldn't coexist between nature and the activities, the industrial activities. So we need to provide efficient anti-fouling strategies, non-toxic, and also to minimize, for instance, the microorganisms who can treatment resistance, because the chemicals are still accumulating in our waters. And so we need to accomplish the European legislation. And there are indeed, these are urgent needs. So what is our intervention? What we are doing to contribute to this universal goal? So we came out with this idea, which is pick up a coating, and instead of following releasing of biocides, which is represented in your left side with these yellow bullets are biocides. So we thought, why not retain the biocide in a coating, in a protective coating? So, but we need to think how to do it, how we can do this. At least we need to maintain the efficiency of the anti-fouling protection, and if we could provide a long-lasting effect, better. So, how we do this? Well, we developed two-step systems. Actually, we pick up a commercial biocide, which is Econaria, which is already used in anti-fouling coatings, but following a release strategy. And we thought, why not using a bridge molecule between the biocide and the polymeric matrix of a coating? So how we can do this? We need to find a molecule that reacts with the function of the biocide, and at the same time have a free one, which is this here, isocyanate, able to link to the polymeric matrix of the paint. So we select these diisocyanate molecules because isocyanate is highly compatible with the matrix of paints or polymeric matrix. Also, this isocyanate is highly reactive with some chemical functions such as this NH group of this biocide. So we move on. We functionalize the biocide and we enable to scale up it. And then we immobilize it in the polymeric matrix, like here in the second step. And for example, I represented here hold the structure for a silicon-based paint. So in your left side, you have the silicon framework, the bridge molecule linked for the polymeric, and from the other side and right, you have linkage to the biocide. We succeeded with this process, and we have the opportunity to have a granted patent already. So, but before reaching all these, I will show you some steps that we taken to prove that it works. So first of all, in the first step, we functionalize the biocide. So we need to prove that this functionalization didn't damage the bioactivity of this biocide. So we performed bioassays with common standard microorganisms in the lab, for instance, Staphylococcus aureus and Staphylococcus faecalis. And from those, we noticed that the pristine cornea and the functionalized one remain quite the same in terms of antimicrobial behavior, or in the minimum regulatory concentration determined. But when we analyze, for instance, for a multi-resistant bacteria, this methicillin-resistant Staphylococcus aureus, we notice a small reduced on their bioactivity regarding the pristine cornea. But even so, it remains bioactive. Also, we analyze that both of them show a bacteriostatic effect, which is quite good in order to attain the treatment resistance from these multi-resistant bacteria. So based on these results, moving on. So in moving on, it means that we go on to the second step. Second step is to immobilize this biocide in a coating and to provide a proof of concept that it works. So here in this slide, I'll show you how we did it. One of the tests that we did it to show it. So we coated ceramic filters here, these ones, one of the ceramic filters, with those coatings, either incorporated or either immobilizing or following this grafting strategy. And so at red and blue one, you will present the controls. I mean, the multi-resistant bacteria culture, and also the coated monolith with the coating without the biocide. And then we have the both ones, yellow and green one, with incorporated or with grafted biocide. And what we can see from here, we can see that indeed our coatings provide a significant inhibition of this bacterial growth. And actually we measured through the number of contact full-informed units, and we noticed that around by 5-fold is lower than the controls. So one thing that you can see here also is that the incorporated Iconae or grafted Iconae have almost the same behavior and steadily a long time. So this also shows us, and after a second test, which is taking liquid salt from the media that was in contact with the coated monoliths, we analyzed that we maintained the bacteriostatic effect on the coated with this quite good. So, but from this, we couldn't even say that our grafted set is better or not related to the incorporated one. So, in order to prove that we improve the immobilization of the biocide coating, we performed these called leaching tests, which are international, following an international standard for the purpose. Basically, we submerged coated prototypes, in this case in artificial seawater, for a minimum of 45 days. And then we analyzed the waters in terms of presence and content of biocides through, for instance, HPLC. And from those waters, we analyzed that when we follow this grafting strategy, the mobilization of Iconae to a chemical strategy, we could reduce the content of biocides in those waters by 10 times more. So it was really a good result. And we were able, by this way, to minimize the environmental impact of these biocidal coatings. So these results motivated us to going further, and going further means that we need to prove the concept at real conditions, and real conditions were provided with the help of storage machines. So we coated prototypes with the most potential coatings, and we were able to submerge these prototypes for more than 2 years. In fact, it was 2 years and a half, 13 months. And we evaluate the surfaces and quantifying them with the percentage affected by biofouling. And we noticed that a commercial coating without our grafted biocide provide after this time 47% of affected area. And with our strategy, we were able to reduce it to 18%. So it was good results and expected in some time, but grateful. So flash conclusions, we are able to graft a commercial biocide in already used marine painting coatings in a silicone framework. Also, we evidence that these coatings provide not only anti-fouling effects in the real world for a long-term period, but also antimicrobial effects in even for multi-resistant bacteria. Of course, this opened our mind to explore to many other applications following this immobilization strategy. And I would like to show where this work was published and all the persons involved on it, and to whom I would like to thank you for all contributions. And you can consult this paper and details of the work available online. And before finalize, I would like to also show you our current activities exploiting this strategy. So we are also able to simulate waterborne flow systems for instance, for water treatment stations with pollutant-based coatings. And using our BioISI mass spectroscopy, we were able to see that for pollutant-based coatings for this strategy, no biocide was detected in water. So this was a really unexpected result. And this work is already submitted. We hope to succeed in the journal submitted. More, we are also exploring this strategy for biocompatible polymer metrics, and also exploring it to many other bioactive agents and bridge molecules, from natural, biomimetic, synthetic, or even we are keen on developing nanoparticles to provide multifunctional activity in terms of photo and bioactivity. Photo, why? Because we also would like to address the degradation of chemical pollutants in water. I mean, it will be great to have a system to in some way degrade pollutants, chemical pollutants from water, and in the other way prevent biofouling. So many current addressing applications, marine infrastructures, water treatment, salinization system, and biomedical in particle states. So thank you. I would like to thank you for your kind attention. Thank you for the opportunity provided by BioISI, and feel free to ask me any question. Thank you.
Okay, thank you, Elizabeth. So time for questions, anyone? Okay. Okay, I have one.
Sure.
I was wondering if this example that you showed, will it be easy to put it on a large scale? I mean, is it easy to synthesize this kind of biocide and then produce it in large scale so you can sell it? And is it economically viable to do so? Because
yes.
some processes might be very expensive and you cannot.
No, yes, indeed, we already done that, okay. But I didn't show. I only show here for this paper because the goal was to do multi-resistance and fouling expect. But we have a prior paper that we test this Econea-based coating in ships, in real ships, through a European project. So we already reached with this coating the scale, the large scale or industrial scale. So yes. We scale up the production of functional Econea and the coating. Just because we use commercial, we use commercial biocide, we use a commercial paint, we just develop a new process. This process is quite simple and feasible to scale up also.
Okay, congratulations.
Well, may I ask you something?
Go ahead, Manuel.
So, thank you, Elizabeth. It was really a very nice talk. But I have a doubt. You know, like the half-life of this biocide, and also like the other side of the coin. We always have two sides. That is, can you be promoting more resistance? So in the sense that your biocide is working now, but maybe in some time in future, all the organisms become resistant.
Exactly. So this kind of biocide is specifically worked for actually hard fooling for the bigger crustaceans. But it is possible. One way that we followed in another paper was to mix biocides to address a big spectrum of action. And indeed, we can mixture. That's why in the last slide I said that we can combine in order to enlarge the spectrum of action. Yes, it is possible. Only with this biocide, no, it's not enough. We need to for instance, we need to use another biocide, which was Irgarol, that we tried to mix with Iconea, following the same strategy. And indeed, it provides a better result because, for instance, Iconea is more for algae, and then the Irgarol is more for eggs and herbicide, and the Iconea is more for hard fooling. So, no, we don't need this one, no. The other question was, sorry, the other question was regarding
the multi-resistant. So if the other organisms can be multi-resistant.
Well, this is a non-releasing strategy, so it works by contact. And since it provides a bacteriostatic effect, I believe that multi-resistance is some kind of overcome for this specific bacteria. I can't guarantee for the others. You need to test it.
Okay, thank you.
You're welcome.
Okay, so the next session was due at 4:30 p.m., but let's make it 4:40, so 10 minutes from now, and we'll meet then, okay? So good afternoon, everyone, and we see each other in 10 minutes.
I wish you all stay because the next session is very lively. It's actually very dynamic and it will be great fun. So all the PhD students are going to make pitches of their work, and I hope you stay tuned because it's going to be great fun. Thank you. See you soon. Welcome back. And this is the most dynamic session of today. So I think it's going to be great fun because we're going to have all the PhD students making pitches about their work. So without further ado, I hope everybody is ready. I'm just going to say their names, and once you hear your names, you may start right away. So we start with Joana Ribeiro, please.
Yeah. Good afternoon. So my name is Joana Ribeiro, and I'm a PhD student at Protein Misfolding and Amyloids in Biomedicine Laboratory under the supervision of Professor Bárbara Henriques and Professor Cláudio Gomes. And today I'm going to present one of our recently published work and part of my PhD project regarding the use of riboflavin supplementation as a therapy for glutaric aciduria type 1 patients. So the next slide, please. Okay, thank you. So glutaric aciduria type 1 is an autosomal recessive disorder of amino acid metabolism that is caused by mutations in the glutaryl-CoA dehydrogenase protein, which is a mitochondrial tetrameric flavoprotein. Currently, the treatment for this disorder consists in a dietary therapy and L-carnitine and amino acid mixtures supplementation. One therapy that is actually been recognized and is widely used for other metabolic disorders that are associated to defects in flavoproteins is riboflavin supplementation, as riboflavin is the biological precursor of flavin. And this therapy bases itself on the fact that flavin can function as a pharmacological chaperone by stabilizing and improving protein folding. However, as this is true for other metabolic diseases, this is not the case for glutaric aciduria type 1. So in order to elucidate whether riboflavin supplementation could be a beneficial therapy for these patients, we investigated the effect of adding flavin to a clinical variant, here represented the GCDH val 400 met. And this variant actually worked as a model for a phenotype associated to severe deflavanylation, as this protein is mainly expressed in its supple form, depleted of cofactor. So by adding exogenous flavin to this mutant enzyme, we showed that the cofactor is actually a driver for structural reorganization, as it enabled the protein to fold back into its native tetramer, which ultimately led to functional recovery. Furthermore, the addition of flavin also improved the variant's resistance to thermal stress, which constitutes a hallmark for the onset of this disease. So overall, our results established the proof of principle for the beneficial effects of this therapy, riboflavin supplementation, in glutaric aciduria type 1 patients. I would like to thank everyone for your attention.
Thank you, Joanna, very much. We have no questions, so if you have questions for Joanna, you may contact her through the chat. And next, we have Lucia Santos.
Hello, everyone. My name is Lucia Santos. I'm a fourth-year PhD student in the BioISI PhD program under the supervision of Professor Carlos Farinha. And today I'm going to talk about CRISPR-Cas9 genome editing as a therapeutic approach for cystic fibrosis. Next slide, please. So as Iris already said on her presentation, cystic fibrosis is caused by mutations in the CFTR gene, which encodes a chloride and a bicarbonate channel in the apical membrane of epithelial cells. Defects in the CFTR protein lead to airway obstruction by thick mucus, chronic inflammation and infections, ultimately leading to progressive loss of lung function. The recently approved CFTR modulators benefit around 80 to 85% of the patients. However, there are still some patients for which there are no effective drugs targeting CFTR protein. CRISPR-Cas9 genome editing could be a solution not only for these patients, but for all CF patients, since it can correct, in theory, it can correct the mutations at the source. So CRISPR-Cas9 is known as the molecular scissors that can cut the double-stranded DNA near the mutation that we want to correct, here in red. And when we provide a donor DNA with the corrected nucleotide, here in green, we end up with a precise repair of the DNA. So we used CRISPR-Cas9 to correct W1282X mutation, which is a nonsense mutation, and we use a human bronchial epithelial cell line. We achieved an editing efficiency of 20%, which increased CFTR mRNA by 20%. That led to an increase in CFTR protein to 12% of the wild type. We are now optimizing a functional assay to assess CFTR function in these edited cells. So that's it. I hope you like my presentation, and thank you.
Thank you, Lucia, very much. Next, Madalena Pinto.
Hey everyone, I'm Madalena and I'm a PhD student from the FungiP group, and my work is focused on restoring airway epithelial function in cystic fibrosis by stimulating alternative chloride channels. Please, the other slide, please. Oh, sorry. Yeah. So as you all know by now, cystic fibrosis is caused by mutations in the gene encoding CFTR, which is an anion channel expressed at the apical plasma membrane of epithelial cells. And very shortly in our work, we studied two alternative chloride channels, namely TMM16A and SLC26A9, that can possibly compensate for the lack of chloride secretion in cystic fibrosis. So for this, we have performed high-throughput siRNA screens using cystic fibrosis bronchial epithelial cells expressing one of these two chloride channels. And we want to identify genes that increase the trafficking to the plasma membrane of DMAM16A or SLC26A9. We are now validating the top hits that can possibly become new therapeutic targets to increase the plasma membrane expression of alternative chloride channels and therefore compensate for the lack of CFTR-mediated chloride secretion in cystic fibrosis. So basically that's it. Thank you. And if you have any questions or if you want to know more, just let me know.
Thank you very much, Madalena. So Romina Coelho is not present due to familial issues. And next is Fernanda Ferreira.
Hi, I'm Fernanda Ferreira from the Cells Tracker and Dynamics Lab, and I'm supervised by Professor Federico Herrera. And today I will talk about Saxine. Okay, Saxine, next please, next slide. The 520 kilodaltons protein Saxine, which is mutated in the early onset neurodegenerative disorder RSAX, includes several functional domains. The presence of the chaperone and ubiquitin-like domains in this sequence and the existing experimental evidence indicate that Saxine could be a functional, could be involved in protein quality control, mitochondrial integrity regulation, and neurofilament assembly regulation, and autophagy control. Mutations in the SAX genes give rise to truncated or defective forms of the multi-domain protein. And we also know that knockout mice shows histopathological neurological features consistent with our SAX. So this is relevant and strongly indicates that our SAX is caused by Saxine loss of function. This opens the possibility to treat our SAX by restoring Saxine function. However, the size and the complexity of the structure of Saxine would be extremely challenging to insert it in patients. So, using cloning approach, we intend to produce smaller versions of Saxine to revert the SAX phenotype at a molecular and at the cellular level. Next, please. Next slide, please. Okay, thank you. For our work, we characterized Saxine expression in all cell lysates from different cell lines, and we were surprised that C6 rat glioma cells expressed high levels of Saxine since they have mixed features of glial and neuronal stem cells-like, and previous reports indicated expression of Saxine only in neuronal cells. Immunocytochemistry of C6 cells shows cytoplasmic distribution of Saxine with some mitochondrial localization in agreement with the previous studies in neuronal cells. Using CRISPR-Cas9 approach, we deleted Saxine from C6 cells and we did the phenotypic characterization of the selected clones. Saxine deletion in C6 cells leads to an apparent mitochondrial depletion and accumulation of the intermediate filaments vimentin, nestin, and GFAP in the juxtanuclear area. These results are relevant because they suggest that glial cells could also be involved in the pathology of the RSAX. The next step of our work will be to restore the wild-type phenotype in our C6 Saxine knockout model using small functional versions of the protein generated for us. Thank you for your attention. If you have any doubt, please contact me. Thank you.
Thank you, Fernanda, very much. Now, next, Rafael Nunes.
Good afternoon, everyone. I'm a PhD student at the Chemistry for Biological Systems group. And I will be talking about halogen bonds in membrane drug recognition. So membrane permeability is a well-known factor determining the pharmacological activity of small molecules. And the process of passive diffusion across membranes is dependent on formation of non-covalent interactions, such as the well-known hydrogen bonds. Also, other interactions may take place, for example, halogen bonds, which I have been studying. Next slide, please. That are interactions where halogens interact with negative sites forming linear interactions. Next slide, please. Halogen bonds are found in protein-ligand complexes or in nucleic acid complexes. So they are, of course, relevant in drug discovery, where many drugs are halogenated. However, there are no reports on halogen bonds targeting phospholipids, which is quite surprising, since phospholipids actually have multiple potential halogen bond acceptor sites. Next slide, please. So maybe halogen bonds play a role in the diffusion of halogenated ligands across membranes. We tackled this doing molecular dynamic simulations. We used, next slide please, as a model membrane, next slide, sorry, as a model membrane of POPC bilayer and several halogenated ligands with different halogen bond propensities. And moving to the first results, indeed, we could quantify halogen bonds in the simulations. They can be up to 25%, as illustrated here, and target both phosphate oxygen and ester oxygen from liquid head groups. Next slide. And in the simulations with iodophenol, which can establish also hydrogen bonds, we found very interestingly that the two types of interactions, hydrogen bonds and halogen bonds, can occur simultaneously, showing that they may act cooperatively. And finally, we were interested in seeing whether if during the internalization process, halogen bonds will take place. So we monitored this a long time in the simulations. And as you can see from the vertical lines, halogen bonds are observed systematically in our simulations. So in the conclusions, we provided for the first time evidence for halogens interacting with phospholipids and for their role in mediating the permeation of small molecules across membranes, which may have implications in drug discovery. Thank you.
Thank you, Rafael. As you can see, some of our students do stretch the 2 minutes to a little bit more, but that's okay. So, thank you, Rafael. Now, Rebecca, André.
Hello, I will present part of my PhD work, effect of Fuchs vesiculoso aqueous extract on cholesterol synthesis and absorption. Next slide, please. You can read the whole work on Food Journal in the publication from the last July, and I am working with the edible brown seaweed, Fuchs vesiculosus, from the North Atlantic Ocean. I start by preparing aqueous extract purified by solid phase extraction, and then I did the LC-MS analysis, and I realized that this extract is rich in fluorotanins and bioactive peptides. As you can see in the next slide, I studied the effect of this extract on cholesterol synthesis because different reports say that different brown seaweeds have a hypocholesterolemic activity. So I studied the inhibitory effect on the HMG-CoA reductase, the rate-limiting step enzyme on the cholesterol synthesis, and I obtained a C50 of 4 micrograms per milliliter. That is a very good result, taking into account that I am working with the complex extract and not with a purified compound. In the next slide, you also can see that I also studied the effect on the cholesterol permeation, and for that I did simulation of the intestinal barrier with KCO2 cells, and I incubate the cells with cholesterol and the extract during 6 hours, and I obtained an inhibition around 45% of the cholesterol permeation. Taking into account these good results, we next want to study the effect of this extract in the presence of ezetimibe, that is the main drug used for the cholesterol lower absorption, and also we want to study the effect of this extract on the proteins that are involved on the cholesterol transport. Thank you.
Thank you, Rebecca, very much for your presentation. Next is Pedro Reis.
Hi, hello everyone. I'll briefly talk about PyPK. That is an important milestone in my PhD plan that is being supervised by Miguel Mosqueiro, Diogo Villaviçosa, and Walter Roquia. Next slide, please. Having accurate pK values for proteins is important on itself, but it's also required in order to predict other properties such as the isoelectric point of proteins or binding energies with small molecules or other proteins. Next slide, please. And to this effect, we have developed PyPK, that is a very fast and easy to use method, and that will become even more easy to use once our online server is released. And as you can see, our PyPK method has a very nice trade-off between accuracy and speed, but we wanted to do better, and to that effect, we have trained our PyPK results with a machine learning algorithm, and we've come up with this PKI model that has the best of both worlds in terms of accuracy and in speed. And with that, I'd like to thank my supervisors and everyone involved in this work.
Thank you very much, Pedro, also for keeping to time. I think we have now a few movies. So the first will be by Flávio Soares.
Good afternoon, everyone. My name is Flávio Soares, and this project is entitled CRISPR-Cas9 Mediated Mutagenesis of SL-GRAS10 Role During Tomato Fruit Development and Ripening. In this project, we are studying the functional role of the SL-GRAS10 transcription factor in fruit development and ripening. Metadata analysis of several transcriptional datasets showed that this gene might be important during flower fertilization or to control tomato fruit ripening process. By CRISPR-Cas9 mediated gene editing, we mutated the gene using two different guides designed and assembled by Golden Bread tool. Heterozygous, homozygous and biallelic plants were obtained, however, and regarding ripening parameters, only mild changes were observed. On the other hand, the number of seeds in some lines were clearly affected. At this point, further analysis is necessary to understand the molecular mechanism behind this phenotype. Finally, we have already developed new double mutant lines to target both the gene SL-GRAS10 and the gene SL-GRAS9 that is resultant from a duplication and may present redundant functions, in order to see if we can introduce stronger phenotypes regarding ripening times. Thank you all, and you can send me an email if you have any questions.
Okay, so Flavio is not online. He cannot answer your questions in the chat, but you can always contact him. So next is another video by Anna Alves.
Hello, I'm Anna, and my PhD project aims to characterize the roles of selected microRNAs in embryo development of conifers, which include species you all know very well, like pines. And there are several differences during embryo development between conifers and angiosperms. But most studies on the molecular regulation of embryogenesis have been done in angiosperms model like Arabidopsis. But conifer models can elucidate the molecular mechanism underlying such differences and at the same time provide information on how to improve somatic embryogenesis, which is a method to induce embryos from somatic cells, and it's used to propagate these species by forest companies. We are using the conifer PCRs as model system to better understand the regulation of embryogenesis by microRNAs. And previous studies on our lab have identified several of these microRNAs with potential roles in conifer embryogenesis, such as microRNA 160. And one of its predicted target is an auxin response factor. And to prove it was the real target of this microRNA, I performed the in vivo validation of the interaction using a co-transient expression of a reported system in Arabidopsis protoplasts. And after a very hard work, I was able to prove the interaction by quantifying the activity of the reported gene luciferase bearing microRNA binding site. And after this, I established the conifer somatic embryogenesis model in the lab, which is working fine. And I am further characterizing the interaction by expression analysis in different stages of PCA embryo development and overexpressing and silencing the microRNA for further phenotypic analysis. Thank you.
Thank you, Anna. I really apologize because I said Flavio was not online, but he actually is. So you can actually put questions to Flavio through the chat and also to Ana Alves. Next, we have another video by Dario Marchese.
Hello, everyone. My name is Dario Marchese, and I'm a PhD student of the Plant Functional Genomic Group. My project title is Functional Studies of Membrane Trafficking and Secretion, Phosphoinositide in Plant Abiotic Stress Responses. We use Arabidopsis thaliana as a model organism to investigate and study the phosphoinositide-related genes involved in membrane trafficking, and more specifically in clathrin-mediated endocytosis, which is a crucial cell mechanism responsible for pore growth, uptaking of nutrients, sensing of the environmental stimuli, and activation of stress responses. We use a reverse genetic approach to study the plant phenotype of Arabidopsis mutant lines, which contain a tDNA insertion in the genes of interest. Firstly, the plant fitness and fertilization rates were assessed by comparing the number of seeds and the calyx length between the mutant and the wild-type lines. Then we focused on pollen cells, which are extremely dependent on polar growth. And by means of in vitro, semi-in vivo, and in situ pollen germination assays, we evaluated the germination rate, the tube morphology and integrity, and effects on guidance. From now on, we aim to study the subcellular localization and the turnover of the proteins encoded by the genes under study through the generation of a fluorescent protein fusion construct and agrobacterium-mediated transformations of Arabidopsis plants. Lastly, the protein-protein interactions will be studied through the biomolecular fluorescence complementation to test spatiotemporal partnerships and regulations. Thanks for watching, and bye-bye.
Now, I think we are ready, and Anna Reis should actually follow.
Hello, good afternoon. My name is Anna. I'm the fourth year of my PhD, and I'm going to share a little bit of my data with you concerning my work in animal tuberculosis field. So animal tuberculosis, can you pass the next slide, please? So animal tuberculosis caused by Mycobacterium bovis is an important infectious disease that affects both livestock and also wildlife species, having an important impact in animal and human health and also economy. In Portugal, Mycobacterium bovis is maintained in a multi-host system with cattle, wild boar and red deer acting as the most relevant host species. However, the transmission dynamics and the role played by each one of the hosts is still unclear. So, in the work that I'm conducting in my PhD thesis is intended to contribute to disclose of the evolutionary and transmission processes within this multi-host epidemic scenario, starting by gaining some insights from the pathogen. So to achieve this goal, first, we perform an extensive work of molecular characterization and Bayesian analysis of over 900 M. bovis isolates from cattle, red deer, and wild boars from a TB hotspot area in Portugal. So we identified 2 TB hotspot clusters, as shown in the map, and we also obtain important information concerning pathogen molecular diversity and population structure. Then, followed this important work, we selected a group that was submitted to whole genome sequencing to perform an analysis at a fine scale, and I'm going to share some key results with you. So first, the SNP-based phylogeny support the branching of the populations into 5 genetic clades with an apparent geographic structure. SNP data together with metadata allowed the study of a SNP network over time, as is expressed in those 4 Portuguese maps. and then the reconstruction of the transmission chains with the establishment of intra and interspecies links with zero SNP differences. We also developed an evolutionary model for M. bovis that pointed the existence of two divergent events in the demographic history of the pathogen in Portugal, and that allowed the estimation of high transition rates between host species that support the importance of wildlife species in the maintenance of animal TB in our country. So this molecular characterization of M. bovis allows us to better understand the flow of the pathogen within this multi-host epidemic scenario, contributing with information to with one piece of the puzzle that is animal TB, and consequently to contribute to propose more suitable interventions and control measures. I would like to finalize by thank you to my supervisors, Professor Mónica Cunha and Professor Rogério Tenreiro, to also the funding agents, so FCT, my doctoral program, and also Projeto Colossus, and thank you for your attention.
Thank you very much, Ana Cristina. And we move on to Diogo Pereira, please.
Good afternoon. I'm a PhD student at Lab Bug Workers in Tech Labs under the supervision of Professor Alan Phillips and Professor Rogério Tenreiro. My project intends to explore the fungal diversity that is inhibiting palm tissues in Portugal, such as leaf spots. The first question one may ask is why palms? It seems to be a reasonable question considering that we are living on a temperate climate. But well, we do like that bold look of the tropics, and palms are undoubtedly a key element on the urban landscape of Portuguese cities. Thus, understanding their fungal composition is a way of managing their healthy usage. Besides this, intensive research showed that palms are an excellent substratum for fungal biodiversity surveys. In fact, palm fungi are a taxonomically diverse group of fungi. These are important not only for the description of novel species, but also for the study of different biodiversity and ecological issues, such as host tissue specificity and co-occurrence patterns. My previous research, which aimed to characterize the fungal communities living on palm leaf spots, allowed me to verify this huge fungal diversity that is colonizing palm tissues. A total of 78 samples yielded around 500 different isolates in 57 different genera, which exhibit a complex community structure with few expected dominant genera such as Alternaria, Cladosporium, and Phoma, and several rare and infrequent genera on the Coelomycetes community. With this preliminary and simple approach, it was possible to detect different biodiversity tendencies, namely in the distribution and composition of the fungal communities, and different ecological patterns, namely in the distribution of and composition of the communities considering different host species. Taking into account these preliminary results, my PhD aimed to address the biodiversity of palm fungi in Portugal in order to understand their functions and dynamics in nature and how they can be used to benefit mankind. Studies will be developed in order to study the composition and structure of fungi, construct different collections where maybe novel species could be described, identify new pathogens and understand their association with the endophytic lifestyle, and search for biological active metabolites. For this, the sampling will be broadened to different regions of Portugal, since in the previous research only some parishes of Lisbon were addressed, and an intensive isolation approach will be taken in order to register the maximum diversity possible. The work plan includes different methods to address the morphological diversity, the genetic diversity through genomic fingerprintings, sequencing and phylogenetic reconstruction, and the screening of metabolites, which will be done following culture-dependent approaches, as well as genomic approaches for the most promising strains. The previous taxonomic studies have allowed our group to already identify novel taxa to science, such as these two examples of a new genus and species in Teratosphaeriaceae and two new marine species, some of which produce these amazing and impressive spores. Thank you.
I can see already in the chat that there are questions among PhD students, and that's very, very positive, because that's exactly one of the things we aimed with this session. So next is Pedro Teixeira.
So I'll just start. My name is Pedro Teixeira. I'm a PhD student under the Microbiology and Biotechnology group. My work was conducted with Professor Rogério Tenreiro and with the biotech company Biotask in the bioremediation area. My focus is on the designing of novel products for bioremediation through adaptive evolution. As you know, nature has to deal with all the pollution our industrial way of life produces, and bioremediation can be a tool to solve those problems. I have focused my research into areas in fog, fat and oily grease residues from food and feed industries, and hydrocarbons produced from combustion engines. These man-made pollutants end up in the wastewater treatment, who rely on microorganisms to eliminate them. But given the stress that our industry and our current systems produce, we must help these systems through biotechnology. Yeah. Thank you. So we use this strategy of adaptive laboratory evolution to find improved and specialized microorganisms by selecting a naturally inocular and submitting it to sole carbon source as a stressful condition. Over many repetitive cycles, we are able to obtain better, more specialized strains than the original ones. This approach allowed me to isolate over 600 microorganisms, which after a profound characterization allow me to select 9 multi-targeted bacteria with several features with better activities, degradative activities, even then compared to commercially available products. Next slide, please. So in the future, we intend to create impactful products which can be used in wastewater treatment to increase the effect of these processes and allow the delivery of the efficient and degradative species strains into our wastewater treatment systems and polluted places. Thank you all for listening, and any questions, just contact me. Thank you.
Thank you very much, Pedro. And next we have João Pedro dos Santos.
Okay, my name is João Santos, and I will be presenting part of my PhD project named Gene-environment interactions in autism spectrum disorder. And I am a student. Okay, so autism is a very disorder. Recent irritability estimates obtained through twin studies suggest that interactions between genetic risk factors such as single nucleotide variants and copy number variants and early life exposures to environmental toxins contributes to the onset of some ASD cases. Among these toxins are phthalates, bisphenol A, air pollutants, and pesticides. Exposures to these toxins are particularly relevant during early neurodevelopment, which is the critical phase for ASD onset. Thus, we hypothesize that early exposure to these toxins can have pathogenic effects in genetically susceptible individuals who carry variants in genes involved in detoxification and regulation of physiological barriers permeability processes, rendering them more susceptible to environmental exposures. Consequently, the objective of this work was to identify variants in these genes in ASD patients. For this, we applied a multi-level integrative strategy, in which large population datasets were inspected for the presence of variants in 519 genes. We found out that 88 genes had SNVs predicted to be pathogenic in silico and or were CNVs unique or more common in ASD subjects when compared to controls. When interrogating the comparative toxicogenomics database, we found out that most of the identified genes interact with relevant xenobiotics. Among these genes are the ones coding for enzymes of the cytochrome P450 family, hormones, and transporters. The presented figure is only for the genes targeted by CNVs, but similar results are obtained for SNVs. These results are important because in genetically susceptible subjects, environmental exposures may be modulated. Thank you very much.
Thank you very much, João Pedro. Next, Joana Vilela.
Good afternoon. I also have a recorded sound, if it's possible to put on.
Let's see if it works.
Thank you. Otherwise,
Autism spectrum disorder is a neurodevelopment disorder with affected brain structure and neuronal connectivity. It's characterized by social communication deficits and restricted patterns of behavior. ASD has a strong genetic component, however, the genetics underlying the disease is incompletely understood. There is evidence that neurotransmission and synaptic biological processes are altered in ASD. So, the objective of this work is the identification of single nucleotide variants in neurotransmission and synaptic genes that play a role in ASD and the biological processes and global networks affected. To do this, we generated a list of neurotransmitter and synaptic genes and identified ultra-rare single nucleotide variants in ASD whole exome sequencing datasets and constructed a protein-protein interaction network and identified the network communities affected. Regarding the results, we selected a list of 1,216 genes as candidates for ASD and identified 358 ultra-rare loss-of-function variants in 209 genes. The main protein-protein interaction network is composed of 59 genes affected by ultra-rare SNVs and is divided in 7 biological communities. The pathways with the best probability inside the 7 communities are associated with pathways as interleukin signaling, G-alpha signaling, MAP kinase, neuronal cell adhesion molecules, phospholipids, and axon guidance. More than identifying ASD risk genes, we are interested in the biological processes affected and the global networks that link those processes together to depict global biological patterns that are common to a larger group of patients instead of analyzing single genes and individual alterations. This work shows that biological processes affected in the cases analyzed are constituted by basic cellular processes that may compromise neuronal development in combination with molecular alterations in pathways more specific from the synapse. Thank you for your attention.
Thank you, Joana. I think we have another Joana. Joana Chora now.
Hello, my name is Joana Chora and I'm a PhD student from the BTR Cardiovascular Disease Group working at Ricardo Jorge. I'll present today the LDLR Variant Interpretation Guidelines by the Clinical Genome Resources Familial Hypercholesterolemia Expert Panel. Anyone working in human genetics is familiar with the American College of Medical Genetics and Genomics guidelines for the interpretation of sequence variants in Mendelian diseases. The Clinical Genome Resource has established expert panels to adjust these general guidelines to specific disease gene pairs. The CliniGen Familial Hypercholesterolemia Variant Curation Expert Panel is composed of international experts working in FH. We defined 2 goals: increasing variant data submission in the 3 genes that cause FH to ClinVar, which is here represented, and they adapt the general ACMG guidelines to the FH context, and I present here the consensus recommendations reached by all panel members. The FHV-SEB focused firstly on LDL receptor gene variants, and specifications include indications for loss of function variant types, functional study criteria levels according to the type of cells studied and thresholds to consider, definition of population data frequency values for benign and pathogenic, specific use and thresholds for in silico prediction tools, and co-segregation criteria specifications. In yellow are the criteria with no disease specifications, and in orange the criteria that requires case-level data. We have just submitted these guidelines for SPI approval, and we have started classifying all 2,800 LDLR variants currently published in ClinVar. We hope that the establishment of these guidelines as the new gold standard in the FH community will help to achieve a consensus, accurate, and standardized method for the clinical interpretation of variants identified in FH patients worldwide in a timely way. I thank you for your attention.
Thank you, Joana. Not sure whether Joana is also online, but we move on to Juan Garcia Moreno.
I'm here online.
That's good.
Yeah. Good afternoon. I'm a PhD student in the Gene Expression and Regulation Research Group. Next slide, please. Well, just to put things in context, when a messenger RNA is degraded, it is usually deadenylated by the CCR4 complex. But sometimes there is a second post-transcriptional modification, which is uridylation by proteases 4 and 7. And here I present our protein of interest, GIS32, which is an exoribonuclease that recognizes and degrades those uridylated mRNAs. Sorry, Finnetin almonds. So we are trying here to figure out the role of GIS32 in cancer. So the first thing we did is to analyze the TCGA database for the gene expression of GIS32 in different tumor types. And we found that in colorectal cancer, GIS32 is upregulated. Next, we did Next, we did an RNA-seq experiment in the colorectal cancer cell line SW480. And we see in this gene expression matrix that under the depletion of GIS32, the overall transcriptome is highly affected. And interestingly, we obtained some Go terms highly enriched for cancer-related processes like cell cycle regulation. Next, please. So that's why at the bench, we wanted to evaluate the impact of GIS32 on cell proliferation by entity. For that, we use two cell lines. The graph above represents what we obtain for normal cells and below for cancer cells. For cancer cells, interestingly, we saw a significant reduction in cell viability. So we wanted to check another important hallmark in cancer, which is cell migration. Next, please. And we did it by wound healing, also using these two cell lines. And again, interestingly, we didn't find any difference when cells are depleted of GIS32 comparing to the control situation in normal cells, but in colorectal cancer cells, we observed a delay in the migration of those cells. So to end, next slide, please. Yeah, so with these results, we can say that GIS32 have a strong impact over the transcriptome of colorectal cancer cells, and it is a potential oncoprotein, given that its depletion reduces cell proliferation and cell migration. Thank you.
Thank you, Juan. As I said, some questions may be put online, also to Joana Chora, the previous presenter. Now, Joana Pereira.
Hello, everyone. I will present part of my PhD with title Stromal Cell-Derived Cytokine Signals Promote the Expression of Tumor-Related Variant RAC1b in Polarized Intestinal Cells. Next slide, please. Inflammatory microenvironment is a tumor-promoting condition that provides signals to which cancer cells respond with change in gene expression. One example is RAC1b. ROC1B is a tumor-related alternative splice variant of ROC1, and it was associated with colorectal cancer. So the main goal of this work is answering the question, do colorectal cells respond to a pro-inflammatory microenvironment with a change in ROC1B, and what is the molecular trigger of this? And for that, we use CACU2 cells as a model. We polarize the cells to better represent the epithelium and co-culture with stromal cells, fibroblasts, monocytes, and macrophages. Next slide, please. By Western blot, we observe that ROC1B is increased when CACU2 cells is co-cultured with cancer-associated fibroblasts and macrophages, mainly M1. By a cytokine array, we analyze the basolateral medium of these co-cultures and identify 4 candidate cytokines, IL-1 beta, IL-6, IL-8, and GM-CSF. And we saw that ROC1B is increased by adding purified IL-1 beta, IL-6, and IL-8, but not with the GM-CSF. Overall, the data is the connection between inflammatory conditions and tumor promoting ROC1B, and may indicate that ROC1B as a potential biomarker of this type of cancers. Thank you.
Thank you, Joanna, very much. We are close to the end. So last but not the least, we have Tanya Marques.
Hi, everyone. Can you hear me? So my name is Tanya. I'm a PhD student in the BioISI PhD program, BioISI. My work is centered around finding an integrative approach to study tissue-specific effects of microRNA regulatory networks. Next slide, please. So microRNAs are endogenous small non-coding RNAs that have a known key role in regulating gene expression. Once they are exported to the cytoplasm, they are bound to the RNA-inducing silencing complex, and then they are ready to bind the microRNA responsive element that is present in the 3' UTR of the target messenger RNAs. Next slide, please. But to complicate the story, we don't have just microRNAs and messenger RNAs. We have other types of non-coding RNAs in the cells, such as circular RNAs and long non-coding RNAs, whose role is not yet fully described, but they are suspected to be acting as sponges for the microRNAs. So this information is necessary to take into account, and also to take into account the tissue-specific transcriptome environment when studying the microRNA action. Next slide, please. To tackle this, my work involves an approach that begins by acquiring public data for paired messenger RNA and microRNA expression in normal tissues, and perform correlation analysis to find negatively correlated transcripts and compare the results with current target predictions. Next slide. The same thing is done at the cell level by looking at single cell expression data and performing the deconvolution of tissues into their cell types, as we can see in the next slide. This allows a better understanding of the cell-specific microRNA regulation. On top of this, this raw data in the next slide would also be used to inspect the presence of other non-coding RNAs, as well as to identify tissue-specific microRNA responsive elements and microRNA sequences. This will provide a starting point for the next slide to build a virtual transcriptome that allows the integration of this information with the binding affinities and retrieve microRNA and messenger RNA interaction rules with the final aim of developing a target prediction tool that takes into account the tissue-specific transcriptome environment. Thank you.
Thank you, Tanya. I think we came successfully to the end. I believe that all our students actually deserve some applause because they did a great job despite some of the technical difficulties. I also hope that you got a flavor of the work, a better flavor because of the diversity of the PhD works which are being developed at BioISI. So now I will just close BioISI Day. I really hope you got lots of ideas, not just among BioISI researchers, so that they foster even more interdisciplinarity, but also with the community of researchers at school and maybe even outside school, because there will be a YouTube video for those who could not participate during the whole day. I think it's a bit heavy to be the full day here, but these are some advantages of these technologies that we pretty much now are using, not for the better reasons, but that's the reality. So I want to thank all intervenants. I want to thank all participants, all the support, the technical support, which was given by Simon Luz from the Secretariat. He was the one sweating in backstage so that things actually happened. Also, Miguel Machuqueiro, all the technical support. A word for the Commission who actually put together the program. I had no intervention, just as a whole. So the Commission included Miguel Machuqueiro, exactly, Ana Nunes, and also Claudio. So finally, I want to thank all the participants who were here listening to the talks, putting questions, and hopefully having lots of new ideas for new projects, new collaborations, and stronger science, not just at BioISI, but also school and in Portugal. Thank you very much to all of you, and that's it.